of around 40 to 100 nanometer in size of endocytic origin secreted by most cell types. They have been shown to be released by cells in response to a broad spectrum of physiological and pathological conditions. And they have been shown to play an important role in cell-cell communication and intercellular transport of molecules. On the right here, there is a scanning electron microscope MDMB 231 breast cancer cell line shown to release certain extracellular vesicles from its membrane. And this is a transmission electron microscopy image of a cell, and it's shown to release extracellular vesicles budding off from its membrane. In addition to exosomes, cells can generate membrane vesicles that are secreted into the extracellular space. Such vesicles can form either at the plasma membrane or in the lumen of internal compartments. Now, this picture shows the most commonly encountered extracellular vesicles in this field. So we have microvesicles, which are much larger in size, and they are mostly released from the plasma membrane. We have exosome-like vesicles. We have early endosomes. We have exosomes, and we have multivesicular endosomes, which can release the exosomes outside. And we have membrane particles. This chart actually summarizes the major types of extracellular vesicles we commonly encounter. Now, this chart is a little old, and I'm sure that there are many other identified vesicles, and there are much better characterization techniques available. But just to have an idea that what are the different types of extracellular vesicles we encounter, and how we can differentiate them based on their size, density in the sucrose, that is sucrose gradient, appearance by electron microscopy, sedimentation coefficient, lipid composition, main protein markers, and intercellular origin. So my work is mostly focused on the exosomes, but there are other, as I mentioned, there are other extracellular vesicles available, and researchers are continuously studying them as well. Now, my talk will be focused mostly on the role of exosomes and tumor microenvironment. Now, these exosomes have been viewed as a source of tumor antigens that can induce anti-tumor immune responses. However, they have been found to possess immunosuppressive properties that can facilitate tumor growth, metastasis, and the development of drug resistance. They have been shown to stimulate angiogenesis, modulate stromal cells, and remodel the extracellular matrix. Now, tumor-derived exosomes have been found to contribute to the establishment of a pre-metastatic niche, which can generate a suitable microenvironment in distant metastatic sites. And another important finding was that exosomal secretion has been shown to be much higher in cancer cells as compared to non-malignant cells. Now, this picture actually summarizes what I talked about. So we have the primary tumor on the left, and on the right, you have the distant metastatic sites and the reservoir sites. So the primary tumor can release these exosomes and can help in the differentiation of the precursor cell, which in turn, the differentiated cell can in turn help in promoting the primary tumorigenesis. It can cause the sprouting or neoangiogenesis of the endothelial cells, leading in the formation of new blood vessels, as we commonly see in cancer. It can release the exosomes, which can inhibit the certain arms of the immune system. For example, it can inhibit dendritic cell, T cell, and NK cell mediated killing. On the other hand, it can release exosomes, which can recruit macrophages and mast cells, and they can actually act as tumor-associated macrophages and help in promoting the primary tumor. The primary tumor can also release these exosomes into the circulation that can go to the distant metastatic sites and create a pre-metastatic niche where the primary tumor can migrate and form a distant metastatic site. It can also cause the recruitment of the bone marrow progenitor cells, which can in turn lead to primary tumorigenesis and metastasis as well. Now, the next question is basically whether the exosomes are functionally active or not. Now, this picture actually shows that exosomes, when released, can be taken up by the recipient cells. So what you are seeing is basically the red, they are the exosomes. And when these HUVEC cells, they are treated with the exosomes, they can take up these exosomes. On the right side, they are showing that these HUVEC cells, once they take up, they can they can be activated. They can activate the signaling cascades. For example, here you can see there is as caspase-3 activation, caspase-9 activation, as compared to no treatment. And here, the HUVEC cells, again, they have been treated with exosomes from the nasopharyngeal carcinoma cell line C6661, where you can see a dose-dependent increase in phospho-ERK activation as compared to no treatment. So they have been shown to be functionally active. Now, why we are studying these exosomes? Now, we know a lot about these exosomes, that they carry a large protein array of, these exosomes carry a large array of proteins, but very little is known regarding the functional relevance of these exosomal proteins, and especially their role in modulating the cellular microenvironment. Thus, having a better understanding of the functional role of the exosomal proteins and how differently the cellular microenvironment works under physiological and pathological condition is very critical. Additionally, identification of specific biomarkers for disease screening, prognosis, and even targeted therapy is also an avenue where exosomal research is currently undergoing. Especially in cancer, the role of the surrounding tumor microenvironment has been hailed as equally important in promoting tumorigenesis as the cancer itself. And in this area, the tumor microenvironment, the exosomes have been shown to be a contributing factor, and thus exosomal research in this field is also critically important. Now, my project depends on one of the exosomal protein, annexin A2, and this is actually, this shows, this website lists the most commonly encountered exosomal proteins as reported by the researchers, and you can see the annexin A2 is one of the most commonly found proteins in these exosomes. And now, a little bit background about what is annexin A2. It's a 36 kD member of the multigene family of calcium and phospholipid binding proteins. It has been shown to interact with negatively charged phospholipids in a calcium-dependent manner. Plasma membrane-associated annexin A2 is integrated in fibrinolysis, exocytosis, endocytosis, cell-cell adhesion, and membrane cytosolical interactions. And more importantly, annexin A2 has been shown to be upregulated in many cancer cell types, including breast, liver, prostate, and pancreatic and ovarian cancer. Annexin A2 lacks a signal sequence that could direct it across the classical ER-Golgi secretory pathway. And the mechanism of the cell translocation of annexin A2 actually was discovered by our lab recently. And it actually has been shown that this annexin A2 binds to the lipid raft region of the plasma membrane and undergoes exocytosis, and thereby can be released in the form of exosomes. And this is actually the previous paper from our lab, where we had shown that annexin A2 can be released in the form of exosomes. And here you can see that when the recipient cells, when they are treated with this exosomal annexin A2, they can be taken up by the recipient cells. And this is the TURF microscopy actually showing up the uptake of this exosomal annexin A2. Some other research groups have reported that annexin A2 expression is higher in the cancer, as I was talking about. So this paper showed that annexin A2 is higher in the media in certain cancer cell types. So here you have MCF10A, which is an immortalized breast cell line. MDB231 is a triple negative breast cancer cell line, and you have two other cancer cell lines, HS578T and T24. All of them, they have higher annexin A2 in the media as compared to MCF10. And this is the quantification of this Western blot. And here also, when you have the MCF10A, which is, as I mentioned, is the immortalized breast cell line, when you have RAS expression and it becomes oncogenic, there is increased expression of annexin A2 in the media itself. So based on all these preliminary data, we hypothesize that breast cancer-derived exosomal annexin-2 promotes angiogenesis and creates a suitable microenvironment that promotes metastasis. Now, we believe that these cancer cells, they can release these exosomes and specifically exosomal annexin-2, which can travel to the surrounding normal cells. And here, this exosomal annexin-2 has some critical functions. It can help in creating a pre-metastatic niche via migration, invasion, ECM degradation, angiogenesis, and endothelial cell activation. And these normal cells then get transformed and they can help in the primary tumor progression. The rationale for this hypothesis is based on the findings that annexin-2 is an important protein component of the exosomes, as I showed you just now. Exosomal annexin-2 levels positively correlate with the aggressiveness of cancer, again, which I showed recently. Annexin-2 is increasingly expressed along with breast cancer progression based on our preliminary data. And once secreted, exosomal annexin-2 is successfully taken up by the recipient cells based on our previous finding. And increased exosomal annexin-2 is responsible for increased angiogenesis and ECM degradation, leading to increased tumor microenvironment signaling, tumor progression, and metastasis. So this hypothesis, we will test this under 3 specific aims. First, we will try to characterize the exosomes as well as the exosomal annexin-2 in a breast cancer progression model. Specific aim 2 will be to determine the role of exosomal annexin-2 in angiogenesis and breast cancer metastasis. And the specific aim 3 will be to validate whether exosomal annexin-2 from breast cancer serum samples can be developed as a potential biomarker. And the cell line models we have used here, as I mentioned, we have used the MCF10 series. Now, this has been isolated from the same breast cancer patient. So MCF10 is the immortalized breast cell line, not a cancer cell line. MCF10AT is a pre-malignant cell line, which was isolated from the same patient. And MCF10CA1A is a highly malignant breast cancer cell line. For the metastasis studies, we have also used MDMB231 and its organ-specific metastatic variants like MDMB4175, which is specifically lung metastatic, and MDMB831, which is specifically brain metastatic variants of this 231 cell line. So for the specific aim, which is to characterize exosomes as well as exosomal annexin-2 in the breast cancer progression model, we will be using transmission electron microscopy, size analysis, exosome release analysis, Western blot analysis, and atomic force microscopy analysis. So these are the electron microscopy images of the exosomes, just to show that we are able to characterize these exosomes, and based on the published literature, we know that we are isolating the exosomes as according to the published and accepted protocols. And they are well below the 100 nanometer size range. And this is the size analysis. As you can see, their average size is around 61 nanometer. And this is the exosome release assay. Basically, it's called the acetylcholinesterase assay, where we looked at the release of annexin-2 in the media among the different cell lines. So you can see MCF10A, which is the immortalized breast cell line, doesn't release that much of annexin-2. The pre-malignant AT releases more annexin-2, but the MCF10CA1, which is the malignant breast cancer cell line, releases the most annexin-2. And this is the Western blot showing the exosomal levels of annexin-2. Here you can clearly appreciate the increased expression of annexin-2 in the exosomal fraction. And these are all the other fractions, which MMP9, matrix metalloproteinase, which are also found in exosomes, plasminogen activator, VEGF, and CD81, which is a marker for exosomes. And this is the Kumasi for the control. And the calnexin actually shows that it is positive for the whole cell lysis fraction, but it is negative for the exosomal fraction. So this actually shows that our exosomal fraction is pure, and we don't have cellular components in that, cell membrane components in that. Now coming to the atomic force microscopy, which usually scans the surface of the exosomes. So we studied the surface of the MCF10A exosomes and the MCF10 CA1 and the malignant exosomes, and we used gold nanoparticle tagged secondary antibody against annexin-A2. So wherever you will have more annexin-A2 on the surface, you will see the attachment of these gold nanoparticles on the surface. So you can clearly see that in the MCF10A exosomes, you don't see much gold nanoparticles on the surface, and this is the topography analysis of the MCF10A exosomes, whereas you can see that more gold nanoparticles are just on the surface of the CA1A, as also you can see from the topography analysis. So this also shows that the surface of the exosomal, the exosomal surface also has more annexin-A2 as compared to the in the cancer cells exosomes as compared to the normal exosomes. And these are our positive and negative controls. CD63, which is the exosomal marker, here we can see that it is positive, and we are using calnexin, which is supposed to be present in the cellular component, but not in the exosomal component. And this is the quantification showing what I was talking about. So in the specific M2, we want to determine the role of exosomal annexin-A2 in angiogenesis and breast cancer metastasis. So for the angiogenesis function, we will use the in vitro endothelial tube formation assay, in vivo matricial plug assay, and we will study the effect of exosomal annexin-A2 on migration and invasion of HUVEC cells, which are endothelial cell, primary endothelial cells. And for these studies, we are using annexin-A2 specific peptides, which is well published. So the LCK-LSL is the inhibitory peptide, and we're using a non-targeting control, which does not inhibit the function of annexin-A2, which is the LGK-LSL. And then we will also study the role of exosomal annexin-A2 in metastasis via tailwind and intracardiac metastasis mouse models. So this is the HUVEC endothelial tube formation assay. So basically, you have a matrigel, and we place these primary HUVEC endothelial cells, and then we treat them with our treatment. In our case, we are treating them with different either PBS or VEGF or with different exosomes, plus minus our peptides. So you can see this is without any peptide treatment. So here you have PBS. We don't see much tube formation. We have VEGF, which is a promoter of angiogenesis. You can see significant tube formation. Here we have the exosome treated from 10A, 80, and CA1A. As you can see, as we go up the ladder, you can see more tube formation as compared to 80 or 10A. And this tube formation effect goes down when we treat them with the annexin-A2 inhibitory peptide, but it doesn't affect much when we treat it with the non-targeting peptide. Now we are not treating the, one thing I wanted to mention is we are not treating the cells directly with the peptide. We are incubating the exosomes with our peptide and then washing them and then treating the cells with those exosomes. We just want to make sure that there is no direct effect of the peptides on the cells. And these are the quantification. So with respect to the mesh formation, so these are the mesh. We use the software ImageJ and count this mesh formation, or we look at the nodes. So this is a particular node, and you can see the branching of the nodes. So this is an ImageJ plugin. We can automatically find out the number of meshes per field. And as I mentioned, you can see there is an increased effect as compared for the CA1A exosome treated cells as compared to 80 or 10A. And the effect goes down when we treat them with LCK-LSL peptide, but not much difference when we treat with a non-targeting peptide. And the same effect is found in 80 as well as CA1. And this is with respect to the branch points per field, and we see the same effect as the number of mesh per field. So this is another confirmation using a different exosomes from the MDMB231. So here we are using SHR instead of the peptides. We're using the MDMB231, and this have the shRNA against annexin-A2. So even in the exosomes, the annexin-A2 levels are less as compared to the control MDMB231 exosomes, and we see the same effect. That with treatment of MDMB231 shRNA, the number of meshes per field and the branch points per field goes down. Now, coming to the in vivo matrigel plug assay. Basically, we use nude mice for this assay, and we mix the exosomes with the matrigel and then inject them subcutaneously on both the right and the left and the right flanks. And then after 10 to 14 days, we excise these. We sacrifice the mice, we excise these matrigels and analyze them. So this is an example of how the matrigel plugs will look. And you can see the angiogenesis, the blood vessels surrounding this matrigel plug. So these are the representative images for the different treatment groups. If we have PBS treatment where we didn't see any blood vessel formation, VEGF definitely is a promoter of angiogenesis, so you can see significant blood vessel formation, and you have increasing blood vessel formation using the breast cancer progression model, which I showed you previously. So 10A, 80, and CA1A, CA1A being the most aggressive breast cancer cell line. And the same effect, and when we add the LC-KLSL, the annexinated inhibitory peptide, the angiogenesis goes down, but no effect with the LG-KLSL or non-targeting peptide. And these are the quantification of the hemoglobin estimation by Rapkin's method. And here you can clearly see that the hemoglobin content of the matrigel plug also is very high as compared to in the 10CA1A exosomal plug as compared to 80 or 10A. And it goes down when we treat them with the LC-KLSL peptide, not much in the LG-KLSL peptide. And this is again with the AT and CA1A plugs. And we also took this peptide and then we sectioned them in paraffin blocks and sectioned them and performed H&E staining. And this is basically looking at the H&E staining for PBS, VEGF. You can see these the blood vessels and the RBCs inside. This is 10A, 80, and CA1A. You can see more blood vessel formation here and more cellular components here as compared to 80 and 10A. And then this goes down when we use the LC-KLSL peptide. Not much difference when we use the LG-KLSL peptide. And this is the immunostaining, just to confirm that these are the blood vessels. So we used PICAM, anti-PICAM antibody or CD31, and this is the DAB immunostaining. And you can see that you have more CD31 positive cells, endothelial cells in the CA1A matrigel as compared to 80 or 10A. And the same finding, so this is 10A, 80, and CA1A. And when we treat them with the peptide, the angiogenesis or blood vessel formation goes down. LG-KLSL, not much difference. And again, we validated it with another breast cancer cell line, NDME231, where I showed you previously that we have the shRNA control, where we don't see much inhibitory effect on angiogenesis. But when we use the annexin-A2 knockdown exosomes, we see decreased angiogenesis, and this is the point of question for that. Next, we studied the role of exosome annexin-A2 in promoting migration and invasion using the transfill assay. So this is the figure, basically, how we use this assay. So we have a transfill and the upper and the lower chamber. In the lower chamber, we have the media, and we put the endothelial cells on the top, and then we either treat them with the exosomes or without the exosome, and then look for the cells which can migrate to the other side of the membrane. If we add the matrigel and then put the cells, then that will be invasion, because the cells will have to invade through the matrigel and then migrate through the pores. And if we don't add the matrigel, then we are just studying migration of the HUVEC cells through the pores. And these are basically the microscopy images of the membrane, and the cells which are migrated, they are stained in purple. So here the cells which have been treated with just PBS, you see few cells migrating. And this is treated with CA1 exosomes, which have the very high the cancer exosomes. And with the LC-KLSL, the number of cells goes down, and with the LG-KLSL, again, you don't see any effect. So basically, whenever you treat with the LC-KLSL peptide, the effect of exosome annexin-A2 goes down. And here you can see that it can promote migration and invasion. These are the quantification for the migration and invasion. Now coming to the breast cancer metastasis model. So for this model, we will use three different cell lines: the MDB231, which is a triple negative breast cancer cell line, and its lung metastatic variant, the 4175, and the brain metastatic variant 831. And we will use three primed groups. So basically what we did was we took this athymic mice, nude mice, and then we primed them. Priming means every 3 days we inject them via tailwind with either PBS or with wild-type exosomal annexin A2 or with down-regulated exosomal annexin A2. And after 1 month of priming, we will then challenge them with the cancer cell lines, which are luciferase positive, so we can track their metastasis. This is just to show that the recipient cells actually, when treated with the exosomes, can take up these exosomes. So this is a red dye which shows the exosomes, and the green is basically the JP positive recipient cells. And this is the model as I was talking about. So when we inject these exosomes into the tail vein, and 24 hours after the injection, and the exosomes have been stained with the red dye, we can sacrifice the animal, take the lungs and section them, and can see that whenever you have exosomes from the cancer cells, you have more exosomes in the lungs. And this is a brain metastatic variant, so that's why we don't see much of a difference as compared to the 231. So exosomes, we can see some exosomes in the lungs, and this is the lung metastatic variant. So basically the exosomes have been isolated from the lung metastatic 4175 cells, and you can see much more, significantly more exosomes in the lungs as compared to the primary or the brain metastatic variants. So there is some organ tropism going on as well. I mentioned that I will be using the knockdown version and the knockdown of annexin A2 for my exosomal study. So basically what I did was, these are the cell line, whole cell lysate, and these are the exosomal lysate, and gap gauge control for whole cell lysate, exosomal CD81 control for exosomal lysate. And these are basically the primary cells without any shRNA, 231 with the control shRNA, and this is 231 with the annexin A2 shRNA. You can see that in the whole cell lysate, the annexin A2 levels go down significantly. And these are the lung metastatic variant 4175, the brain metastatic variant 831. And these are the exosomal component from the same cells. So basically we collect the media from these cells and then perform isolation and then get these exosomal components. These are the 231 wild-type exosomes, 231 control exosomes, 231 SH and X-ray2 exosomes, 4175 and 831. So this also shows that whenever you knock down with shRNA, you see a significant decrease in the exosomal component annexin A2 as well. And so for this model, we used, as I mentioned, the regular priming, and that, as I said, every 3 days we will inject the tail vein. We will prime the animals with these exosomes or PBS, and then after 1 month of challenge, after 1 month of priming, we will challenge them with the breast cancer cell lines. And we use two different models. One is the lung seeding, where we are injecting via tail vein, or we inject them in the left ventricle of the heart, which is the intracardiac injection model. So for this one is it's like experimentally induced metastasis model. And this is the pipeline we have used. So basically, we take athymic nude mice, and then we challenge them, and then after 1 month, we inject with the cancer cells. And then we perform bioluminescence imaging every week. And once considerable metastasis has been observed, we euthanize the animals and organs are harvested and studied further. So this is the first, I will talk about the lung metastasis. So this is basically, we prime with exosomes and then challenge them with the MDB231 luciferase positive tail vein injection. So we are seeing only the lung metastasis in this, and this is the India ink stain showing, so whichever you have the tumor, wherever you have the tumors in the lungs, those will not take up the stain. Those will come as white. You can see the nodes here. So this is the PBS primed 231 shRNA exosome and then 231 shRNA control exosome. And you can see significant tumor formation in the shRNA control exosome, but it goes down when we use the shRNA with the annexin-A2 against annexin-A2. And this is the photon flux, basically looking at the luciferase signal over time. The same, same model, but we used the 4175, which are lung metastatic variants. So we injected in the left ventricle of the heart, as I mentioned. And after 44 days after the challenge, so this was 35 days, and this was 44 days. Again, we looked at either the PBS primed or 4175 shRNA annexin-A2 primed or shRNA control. So basically, the idea is when we are priming these animals with these exosomes, we see increased metastasis. But when we priming, there is a difference between priming with shRNA annexin-A2 exosome or shRNA control exosome. So when we are priming them with the shRNA control exosome, we see even more increase, further increase in the metastasis as compared to when we are knocking down annexin-A2 in the exosomes. And this is again the bioluminescence imaging showing that 4175 control exosome primed animals have significantly higher metastasis, lung metastasis, as compared to the knockdown exosome primed animals or as compared to the PBS. And these are the H&E staining and the DAB immunostaining against human Biomedical. And you can clearly see that the lungs, these are mouse lungs, and you can see the tumor cells here. So not much of micrometastasis in the lungs when we have the knockdown annexin-A2 exosomes primed animals, and you have a big metastasis in the lungs, as seen with the tumor mass when you have the control exosomes from the primed animals. And this is the same with the Biomedical, showing that whatever cells we are seeing, these are actually human cells and not the mouse cells. And now coming to the brain metastasis model, where we used again two different models. We used the primary MDB231 cell lines. So here we are only doing the IC, intracardiac injection, because we are trying to see the brain metastasis model. So this is PBS primed, knockdown shRNA exosome, knockdown annexin-A2 exosome primed, and control exosome primed animals. And we can see that when we are priming them with control exosome, it's significantly higher brain metastasis as compared to knockdown annexin-A2 exosome primed or PBS primed. And this is the luciferase quantification for that. And this is with the 831, which is preferentially brain metastatic variant, and you can see much more brain metastasis in the control exosome primed animals as compared to knockdown exosome annexin-A2 exosome primed animals or PBS primed animals, confirming what we saw with the other experiments as well. And these are the sections from on the left. You have the H&E sections of the brain, and you can see micrometastasis in the brain, in the mouse brain, and this area you can see more micrometastasis. And again, this is the DAB immunostaining against human Biomedical, showing that basically these are the human cells, because the MDB231 are human breast cancer cell line against the mouse background, mouse brain, and these are again the human micrometastasis on the breast cancer cell line. And this is the time course of the organ-specific metastasis, basically looking at the photon flux using the luciferase signal. And then there's the 4175, and there's the 831, the lung metastatic and the brain metastatic variants. And you can see wherever we are knocking down exosomal annexin-A2, we are seeing a decrease in the tumor metastasis progression, either in the 4175 model or 831 model. Now, so basically, we believe that, or we hypothesize that this priming with exosomal annexin-A2 creates a microenvironment conducive of promoting metastasis. So we wanted to see what signaling changes it is this priming with the exosomes with normal or knockdown annexin-A2 is causing. So we took these HUVEC cells and then we treated them with either PBS, or with 231 control exosomes, or so this is from MDMA 231 breast cancer cell line, or 231 shRNA annexinated exosomes. And we found that whenever we have whenever we are treating these cells with these exosomes, the signaling goes up. My bad, actually. This is supposed to be control, not annexinated. We can see this here. Whenever we have the control exosome prime cells, we have increased in phospho-NF-kappa B, phospho-P38, and phospho-STAT3. And whenever we knock down annexinated levels, we see decreased phospho-NF-kappa B, phospho-P30, and phospho-STAT3 signaling in the civic cell lines. We wanted to see that whether this signal, we tested multiple signaling molecules, and we found these were upregulated with exosome treatment. So we wanted to see whether from our animal studies, whether we can see the same finding. We can see the same findings. So what we did was, from our animal studies, we took these lungs and looked at these molecules in the DAB immunostaining, and we found the same finding, that wherever you have control exosome treated animals, we have more staining of phospho-STAT3, phospho-NF-kappa B, MMP9, and phospho-P38 as compared to knockdown of annexinated exosome treated animals. So more brown means more staining for these molecules. And this is from the brain sections. We found the same thing, that we have more brown stains of this phospho-STAT3, phospho-NF-kappa B, MMP9, and phospho-P38 in control exosome-treated animals as compared to the knockdown exosome-treated animals. So the possible mechanisms, what we believe, this activation of phospho-P38, NF-kappa B mediated pro-inflammatory cytokines and chemokines, they are recruiting the tumor-promoting macrophages. And it has been shown recently that annexin A2 can activate the macrophage for the IL-1, IL-6, and tumor necrosis factor alpha secretion through the TLR4 and MyD88 and TRIF dependent pathways. Another finding, we also found that exosome priming can also activate VEGF-R1 in primed animals. Now, this is not based on the annexin A2, it's just by priming. As compared to PBS, if you prime the animals with this MDMA 231 exosomes, you will see increased VEGF-R1, VEGF receptor 1 signaling in the lungs or the brain, which is also significant finding. Now, having talked about the in vitro and the in vivo effects of these exosomes and exosome annexinated, I wanted to further validate whether there is any potential of this exosome annexinated as a breast cancer, as a potential biomarker for breast cancer patients. So we did a pilot study where we assessed normal age-matched normal control and breast cancer serum samples. So this is the ELISA. On the left, you have the whole serum annexinated. On the top, you have the whole serum annexinated. You can see that this is normal and this is cancer. This is the absolute value of serum annexinated, nanogram per ml, and this is the fold change related to normal. And in the bottom, you are looking at the exosomal component of annexinated, and this is the absolute value, and these are the fold change. So you can see that the annexinated is much more concentrated in the exosomes in the serum sample of these patients. And this, again, we looked at the serum exosomal annexinated levels, a nanogram per ml, in different patient samples. So breast cancer patient samples based on the tumor grade. So we have non-cancer patients. We have tumor grade 1, 2, and 3, and you can see there is increasing serum exosomal annexinated levels as the tumor grade increases. And here we also correlated that with the overall survival. So whichever patients had higher exosomal annexinated levels, their overall survival was poor as compared to patients which had low serum exosomal annexinated level. And this is disease-free survival, again showing the same thing, that wherever we had higher serum, whichever patient which had higher serum exosomal annexinated levels, they had poor survival as compared to patients which had less exosomal NXN2, serum exosomal NXN2. And here we also studied the serum exosomal NXN2 level. This is the serum NXN2 level. This is the serum exosomal NXN2 level. We were lucky to have some HER2 positive and triple negative breast cancer serum samples, and you can see that serum NXN2 itself is very high in HER2 as compared to normal, and even higher in case of TNBC as compared to HER2 or normal. But when we looked at the serum exosomal NXN2 in non-cancer, ER positive, HER2 positive, and TNBC, we saw a significant difference among the different breast cancer subtypes, being TNBC having very high levels of serum exosomal NXN2 levels as compared to HER2 positive or ER positive, which all of them were higher than the non-cancer. So it can be, so this was a pilot study. So we are still studying to see and expanding and trying to see it in more patients and trying to see if this can be expanded to other patients as well. So in conclusion, I would like to say that hopefully I was able to convince that exosomal NXN2 is expressed in higher levels in safety and CA1A, which is the triple in the breast cancer cell line as compared to the pre-malignant or the immortalized breast cell line. And we did that with by showing by studying them via electron microscopy, doing force microscopy, Western blotting, using the 10-year progression model. And then we determined the role of exosomal NXN2 in angiogenesis and breast cancer metastasis, and we used the in vitro endothelial tube formation assay, the in vivo meat-rigid plug assay, the Hewitt migration and invasion assay, and then we used two different injection models, the tail vein and the intracardiac injection model. We found that exosomal NXN2 promotes angiogenesis. It can promote migration and invasion of the pubic cells, and cancer exosomes are capable of promoting organ tropism, and exosomal NXN2 promotes breast cancer metastasis to lung and brain. And we also showed using mouse and cell lines that exosomal NXN2 can prime and create a microenvironment conducive of promoting metastasis by activating the phospho-P38, NF-kappa B, and STAT3 signaling cascades. And we found that just the treatment of cancer exosomes, it can lead to VEGF-R1 upregulation in parenchymas. And in the third phase of the study, we looked at the exosomal NXN2 in breast cancer serum samples as a potential biomarker. So we had breast cancer samples, and we had serum samples and age-matched normal samples, and we found that the exosomal NXN2 positively correlates with the aggressive subtypes of breast cancer as seen in a pilot screening of breast cancer and control serum samples. I would like to conclude my presentation here, and I would be happy to take any questions you have.
Lecture at the 41st Annual Conference of IABMS on polyherbal formulation of ISM drugs, IBMS, University of Madras, and Dean UICIC. And currently, I am Director, Dr. C.L. Biomedical Foundation for Pharmaceutical Education and Research, Chennai. My topic is polyherbal formulation of the Indian system of medicine. This ISM is also known as the Indian System of Medicine. It stands for the Indian System of Medicine. WHO has recognized the Indian traditional system of medicines like Ayurveda, Siddha, Unani, and homeopathy under the World Traditional Medicine domain. And Government of India has got separate independent ministry known as Ayush under the Ministry of Health and Family Welfare of Government of India. In India, but all over the world, it amounts to about 1,200 plant-derived medicines are used in the Western medicine, including functional foods and nutraceuticals. Herbal medicine in the form of old plant plant extracts and the parts of the plants are used as a main constituents of the Indian system of medicines formulation, both Shastric medicines and proprietary medicines. The proprietary medicines means that they are different from the Shastric medicine, but they are prepared by using the research on the products, herbal products. They compound it and make a formulation to treatment of the diseases according to the nature of the pharmacological action of the individual drugs. There are more than 2,500 oral formulations available in the Indian system medicine pharmacopoeia, covering Shastric and proprietary herbal medicines. And many traditional practitioners use even single herbs also for the treatment of many diseases. Western medicine practitioners criticize the polypharmacy, that is more than one drug entity in the formulation. I think they have misunderstood about the ISM polyformulation. It is not a polypharmacy, it is a polyformulation. The formulation means that contains many drugs in one single preparation, whereas polypharmacy means a man takes different drugs for the different diseases. That amounts to the polypharmacy, especially a man who is hypertensive, who is also diabetic, and who is having hypercholesteremia. Probably in Ayurvedic medicine, he will be taking about 8 drugs per day. That is a polypharmacy. But it is required, according to the modern medicine, these 3 drugs are required to control the diabetes and complication of the hypertension, or also to treat the hyperlipidemia and other things. But in Indian system of medicine, these drugs, 1 or 2 or more than 10 or 20 drugs are, herbal molecules or herbal parts are, mixed together, and they make a extract, or they make a solutions, or they make a powder, and it is given as the one drug, and it may contain more drugs, but you cannot call them as a polypharmacy. Now, there are 2 types of formulations are available for the ISM drugs. One is the Shastric drugs, usually reference taken from the formula derived from the ancient textbooks of the Indian system of medicine. See, for Charak Samhita, Bhava Prakasha, Sahasra Yoga, Tirumandaram of Tirumular for the Siddha medicine, like that, for each system of medicine, they have got a references are available in the textbooks written by the Pandits, or those who are investigate this system about 5,000 years back, and that is written in the palm leaves, and that has been translated or taken and then digitalized by the Ministry of ISM to get more popularized about the system we are practicing earlier. So these systems, they are known as the Shastric drugs. The regulation to obtain license for manufacturing these things are different from the license conditions for the manufacture of the proprietary drugs. So proprietary drugs is another type of formulation that is being used by the manufacturing companies, but they are not pure, Ayurvedic, but they are manufacturing, even modern medicine also they will be manufacturing. They will be having a separate division to manufacture these ISM drugs. That is, formulated by a scientist or formulated by a physician from his experience, and he said he prepares a different formulation that is known as the proprietary medicine. So, to get a license for the proprietary medicine, you have to submit some data, just like any research required. Any research that is being done, even for vaccines, the clinical trials should be conducted, preclinical studies should be conducted, toxicity evaluation should be done. All these parameters to be observed when you are applying for the patented medicine or the proprietary medicine. So these 2 categories are there. One is the Shastric and another one the proprietary medicine. Then, what is the, standard, reference, standard, the pharmacopoeias are there for modern medicine. There are Indian pharmacopoeia, USA pharmacopoeia, and British pharmacopoeias are there. Pharmacopoeia is the compendium of all the, properties of the drugs, their classification, their chemical structure, identification, how to analyze the components present in this formulation. For that, I think there is a standard method of analysis and standard methods of evaluation of the quality and the quantity of the main ingredients is added, what are the excipients are added, what are the things that are not to be added are present in it as a spurious drug, or it is as a contaminant, or it is maybe a adulterant. So to analyze these things, I think to find out there is a standard book, there is an SOP, just like an SOP. If you get a medicine to be analyzed, that you have to do it, or you have to mention what is the pharmacopoeia standard of the drug you are used in the formulation. When paracetamol IP or paracetamol USP, you might have seen it in your allopathy drugs, like that, but not in the, for Ayurvedic drugs or ISM drugs, there is, only they have to mention from which textbooks of the Ayurveda you have given this reference for this drug only to be given. For that, they have recommended so many books, a selected number of books are there, or selected number of publications are there. All those things are put together in a nutshell. That is the pharmacopoeia of ISM drugs. So just like the pharmacopoeia for the modern medicine, Indian system of medicine also has got a pharmacopoeia lab, Ayurveda pharmacopoeia, Siddha, Unani, and homeopathy pharmacopoeias are there. According to this pharmacopoeia, wherever they are in India, whether it is in Trivandrum or in Ghaziabad, the standard of the drug should be same as far as the pharmacopoeia standard is concerned. The identification of chemical characteristics of the active principle to be used as a marker and a host of other information from raw materials to finished product. Here, the selection of the raw materials for the preparation of ISM drugs is really a strenuous process, because it is cultivated from the wild, or it is, taken from the wild forest and other things. Somebody should identify it is the proper drug that has been taken for the preparation of the formulation. So there are, some methods according to the WHO GMP, good manufacturing practice of the Ayurvedic, Siddha, Unani, or homeopathy drugs. The GMP, that is the good manufacturing practice, should be followed. For that, they have given so many, parameters to be observed before starting the preparation of the drug with the raw drugs. From the standardization of the raw drugs, or from the standardization of the selective extract of the raw drugs, all those things should be standardized, and that also should be present in the pharmacopoeia. According to the pharmacopoeia standard, you have to prepare these things. This GMP practice is mainly, followed now, because India is getting very good, export potentials for our drugs to the Western countries. So Western countries, those who are importing Indian medicine, they insist many things to be checked at the manufacturing side, the manufacturing processes, and you have to give a good GMP as well as also standard operational procedures for the preparation of the drugs. They insisted if we change our policy, to make a little more money, then the consignment, if it goes from here and they found the standards are not good, they will reject it. The standardization, there are so many issues are in the standardization of the herbal medicine. The foremost is that it is not a single. See, the formulation contains maybe 10 or 20 or 25, 30 ingredients. Ingredients means the herbs and the related products of the herbs will be there. So it is very difficult task to standardize the herbal medicine preparation. A single drug, if it means a single drug. For otherwise, you take for a paracetamol syrup, or you take a ibuprofen syrup, or you take a metformin. All these things are only single ingredient, and the single ingredient is very easy to get a standardized analysis. And you can use an HPLC, or you can use a sophisticated LCMS, or whatever it may be. And even in the very small quantities, you can examine whether the preparation contains so much amount of the material is there. So it is a very easy task, and it is the standardized procedure, and it is followed well all over. Whereas the ISM drugs, it contains more than 10 or 15 drugs. So identifying individual drugs is humanly impossible. But only certain methods you can use it only, the markers. Because we are, aiming the treatment with the one single drug or, many drugs incorporated into that gives the beneficial therapeutic effect. So, that means that the marker, that is, suppose you take an alkaloid as a marker in a take a flavonoid as a marker in the particular preparation for anti-inflammatory activity or antioxidant activity, then that will be the marker, and you standardize on that marker plus any drugs that are, very quite relevant for the particular disease, that the efficacy is more, or it will be increasing the efficacy of the other one. You come to know that, or by research or by experience, that marker, that particular marker also you can include in the analysis of the drug, of the finished product. So, but it is very difficult process. That's why many times, we are, making effort to, standardize this. Sometimes the prepared extraction doesn't contain the quantity of the marker is there. When you examine, the marker will not be found out because the method is not, highly relevant. In the sense, I think very difficult to, get the not, very, very small quantity of them present in the mixture is very difficult to, identify. So that determines the amount of the active principle or the marker is present. Accordingly, you have to decide an analytical system to find out that much is there or not. Otherwise, it will be rejected. Then identification of a particular constituent when the mixture is practically impossible by physical means as well as also chemical means. Then another problem is the identification of the genuine. What do you mean by genuine drug means? It is again another problem of genuine drugs. See, you take for example Phyllanthus amaris, Phyllanthus nirudi, and so many Phyllanthus subclasses are there. Species are there. Suppose you are all of them morphologically look like similar, but only a systematic botanist or you as an herbal botanist can identify it, and you can say this is amaranth or this is nirudi or something like that. Just like that, there are so many problems to identify the authentic medicinal plant. If the authenticity of the medicinal plant is not correct, the therapeutic efficacy of the mixture of the compounds or mixture of the formulation may not be as good as the other batch which contain the authentic sample. So it is very difficult. So you have a reference standard of the authentic drug. That means your laboratory standard, because there is no national standards of that. Only laboratory standard is there. Then you consider your laboratory standard is the best, or for your manufacturer, because you have found out your products are good, your products are giving desired pharmacological as well as therapeutic action. You think or you standardize that your raw material that you have selected is the right one. But in textbooks also, they didn't say, they give it only in the vernacular name, not in the botanical name. So it will be very difficult touch to identify. But the pharmacopoeia of Indian medicine system of medicine, Ayurveda, Siddha, now they have brought about the monographs of the each and individual plant, and accordingly, about the adulterants. If the genuine drug is not available, or there is a possibility to obtain this genuine drug from the market, there are substitutes. Sometimes these substitutes are, allowed in the textbooks of Ayurveda and Siddha Unani, provided the genuine drug is not available or the genuine drug cannot be recognized by the man who is, doing the selection of the plants. And in that place of genuine drugs, to compensate the particular drug, there are substitutes are there, or they can use spurious drugs. The spurious drugs is the biggest problem, because even substitution also is not allowed, because the substitution is also come under the category of the spurious drugs or adulterants. So when they identify these spurious drugs or adulterants in that formulation, then it will be rejected because the adulterants will give you harmful effect than a beneficial effect for the patient. So even a species difference is also make one drug more effective, and sometimes it will make more poisonous, or side effects will be more with the spurious drug. Another point is about the drug-drug and the drug-food interactions. This drug and drug-food interactions now is being recognized by modern medicine, and they will always, say about that. If you take Ayurvedic drugs, you have got a lot of restrictions on your food, a lot of restrictions on your drinks. All those things are there. But when you take an allopathic drug, there is no such restriction. Now they also change their tone now, because allopathy also says that there is a drug-drug interaction and drug-food interaction. See, the drug-food interaction, whether it is good or bad, that depends upon the interaction that takes place, and it affects the ADME profile, that is absorption, distribution, metabolism, and excretion profile of a drug. Sometimes it will increase the bioavailability of the parent compound or a foreign marker from the allopathic Ayurvedic drug. And sometimes, it will decrease the absorption of the drug. So what will happen? I think we cannot, pinpointly say whether the patient has got the enough drug according to the drug regimen. So you give an example, curcumin. Now, curcumin is available in different dosage form are available. And all the manufacturers, they say our curcumin is superior because it is water soluble, it is easily absorbable, and its bioavailability is good by manufacturing and using different techniques to get a formulation, all those things. So this drug and drug interaction, that is an Ayurvedic drug versus an Ayurvedic drug, or an Ayurvedic drug versus an allopathic drug. It is a very common, and the physicians of modern system of medicine now, they also advise you not to take Ayurvedic drugs along with the allopathic drugs, because the interaction may be due to the tannin that is present in the, lot of tannins are present, and other alkaloids are there, flavonoids are there, terpenes are all there in the Ayurvedic medical formulation. Sometimes they may interact with the modern, the modern entity, the allopathic moieties, and it will reduce the absorption, increase the absorption, or change the metabolic pathway or metabolism itself. And so many factors are there. And that's why drug interaction and drug-food interaction is an important factor to be decided while taking the allopathic and Ayurvedic formulation along with the allopathic drugs. The polyherbal nature of the ISM drugs. And people may not accept this theory, because a polyherbal means it contains the plant's parts, that is flower or bark or root or leaves, whatever may be the parts that is used for the preparation of the ISM drugs. They are all not from the same family or same species, maybe from different families and different species, and also contain different chemical constituents like flavonoids, alkaloids, terpenes, coumarins. All those things are, different constituents will be, chemical constituents will be there. So what is happening here? I think they may interact. This principle of interaction, drug interaction may occur, but it is judiciously compounded or formulated from the long-standing experience of the practitioners of the Indian system of medicine. They have empirically observed these things, and they are treating the patients of their community, and they know the demography of this community. They know the diseases prevailing in that particular community and which drugs are useful for the treatment of that disease. And it is a closed community, so they can interact each other and find out and record whatever they observe and they were told about the action, reaction, side effects about the drugs. It is about 5,000 years old system. So there is no record. That is the only problem. It is verbally communicated from the guru to Shishya, and the Shishya will be communicating to each Shishya like that. So there is no records. But now it has been improved after the councils are started for the Indian system of medicine. They have started research products on the various aspects of the interaction and other things. So what is happening here? These things should be always observed. Why it is judicially have been compounded? So you can see one alkaloid will be there, and one plant, and another may be contributing more, flavonoids. So generally speaking, these alkaloids are bitter principle, flavonoids or flavonoids, glycoflavonoids, all those things are, little bit different taste and this thing. Their action also may be different. So in that way, if you take it, considering the nature of the chemical moieties present in the different plants constituents, and the amount of these constituents present per gram or per kilogram in a batch, that will be determined by them. But the one advantage earlier was that they can identify the plant or plant species from the wild. Now, you don't have any opportunity to get these plants from the wild. Either it is cultivated, in a particular area of the land, or sometimes they are picking it up from this thing. But you don't know what is the condition in which it is grown. Is there any pollution is there? Suppose you take, there is Spida cordifolia, there is one drug is used for the preparation of the Dasamularistam. When you take it from the roadside, it contains a lot of lead because of the pollution that is emitted from the motor vehicles. So there are situations in which these were, rejected by the importing Western countries, selling that preparation contains a lot of lead. The manufacturer has not added any lead by its conscious. Will not say that you should add lead, or they may say that it might have, leached out from the machinery instead of, using this old machinery. The lead might have leached to the solution or something like that. They may say, but it is the fact that lead is present, and they have collected the medicinal plant not from the isolated place or from a wild. It is from the roadside. It contains a lot of lead in that. Like that, many things are there's a problem for that. So this problem will persist unless you have got for a herbal gardening or herbal big scale manufacturer cultivation of the herbs. And how to avoid these things is the only method. Community, it should be those who are engaged in this drug manufacturer as well as also collection of the drugs should be trained properly, and also encourage the farmers in a particular locality where water is not required much, or the climatic condition is very good for a particular type of herbs to be grown. All those things should be taken into consideration and engaged. That's the national here, the herbal gardens. Dr. Shastri will be talking about the activities of the national herbal cultivation, and they are giving grant for the agriculturists and other people to start this thing. If we have got such a standardized agriculture, farming technique, cultivation, manuring, harvest, storing, all these particular system is standardized, definitely we can improve our standard of the manufacturing of the ISM drugs. Plus, we can remove or eliminate the adulterants as well as the spurious drugs in the preparation. Then, in such a way that it has got a agonistic or antagonistic action, means if it is appeared, the compound, that formulation may contain a drug which will take care of and reduce the side effects. And if it is a poisonous or an overdose, it is producing some effect, then there will be antagonistic or it's an antidotal action of one drug will be there to reduce it. So it has got benefits and counteracting effect, addictive, potentiation acts, and all put together in the different dose. All ingredients are not in the same dose. Different doses are there. The main pharmacologically and therapeutic active principles will be on a higher level dose, and other things that are put it into the composition or the formulation will be on a very, very small little dose. So in the small little dose per se, they don't have any action, but it can act as a addictive or a potentiation or enhance the bioavailability or reduce the toxicity type drugs will be there in the formulation. I think all, you cannot, there are pros and cons. The benefits are there and the demerits are also there. The one main thing is the difficulty to identify the precise mechanism of the herbs responsible for therapeutic action. Here, we explain that is evidence-based medicine. Modern system is the evidence-based medicine. You have to prove it. What is the mechanism of action? Unless you prove the mechanism of action and evaluate the toxicity, long-term toxicity and acute toxicity, all these are, fundamental principle, a good practice, of the allopathic medicine. That is standard practice. But whereas in polyherbal formulation, the main problem is what is the mechanism of action of the main drug or the drugs that are added in a little quantity in a formulation. We don't have any, method to identify it. Now, some laboratories they are doing now, pharmacopoeia of Indian medicine system also they are doing some research. Institutions are doing research, and Ayush is financing for the research for the bioavailability studies as well as also mechanism of action studies, the pharmacokinetics and pharmacovigilance. All those things are now carried out for the Ayurvedic medicine. Then the second is the difficulty to standardize the dose requirement. Because the dose requirements, according to the active principle that are present in it, and we are also doing some work on the standardization of the marker. If that marker is up to this mark, then the concentration of the active principles will be like, we are concluding in that. But a dosing is also a very big problem, because in allopathy, the dosing is, structurally, and it is, very clearly evaluated by the pharmacokinetics, that is absorption, distribution, metabolism, and excretion. All these 4 or 5 principles, I think they are observing, and they will know how long the drug stay in the body, how long it will stay in the body, and what is the mechanism by which the drug's potency is reduced, get it metabolized to a different, or get it metabolized to another compound which is more, beneficial than the parent compound. Or when it get it metabolized, whether the metabolites are excreted or does it stay in the body and produce toxicity. But all these things, kinetics are well studied. So there is no dispute about that. But the kinetics of our herbal medicine formulations are, not in the only initial stage or in the childhood stage. Then the difficulty to identify the herb responsible for the side effects also. Which is responsible for the beneficial effects, is very difficult to identify in the polyherbal. And it is also the same thing to identify which drug is more producing side effects also. Whereas in allopathy, that question also is well established, well studied, and when you come the complete dossier about that particular moiety or a chemical entity, it is, in the pharmacopoeia or for the promotion, for the promotion of the product by prescriptions, everything is given, A to is given. What is the, milli equivalent of this will be there in the body, milligram will be there. If the milligram, how much milligram is required to maintain the fever, just like paracetamol, every 6 hours you take 500 ml or 650 paracetamol you take every 4 hours. Like that, this is, accurately, it is, standardized in allopathic drugs. Now, that standardization is not in the, ISM drugs. The effort should be taken to identify, and thorough study of the kinetics is a must, to get more information about the beneficial effects as well as also the dosing, duration of treatment, and so many other profile of the pharmacokinetic and pharmacodynamic profiles of the drug molecule. Then difficult to identify and prescribe the specific antagonist drug to prevent the morbidity and mortality. Then if you take a drug, I mean, that is a polyherbal formulation, you may get, I mean, sometimes the patient who receives may be allergic to a particular ingredient in that product. It is a plant material. So what will happen? Sometimes some of Indian system of medicine. Hope I think you have understood. It's only covered superficially. I have covered. I mean, presentation about that. If you have got any serious problem, you have got any problem to be discussed, you are welcome to discuss personally. And my ID and other things, I'm a member of the Indian Association of Biomedical Scientists. You can get me from the secretary and you can have a dialogue with me. And thank you very much.
Hello.
Yes, please start, sir.
Oh, talk. Okay. Welcome, friends. This topic I have taken up, even though it is known for a long time to everybody. Among the nutrients that we take in our diet, proteins are very important because the proteins are made of different amino acids. Our body contains proteins which are made of 20 different amino acids. Can I have the next slide? Can I have the next slide? Okay. Now the different amino acids that we have to take in the diet are called dietary essential amino acids or simply essential amino acids, but I don't call it essential because every amino acid is essential. So those amino acids which are essential in the diet are threonine, tryptophan, methionine, isoleucine, leucine, lysine, valine, and phenylalanine. There are 8 amino acids in this list that I have listed. And these 8 amino acids we have to include in our diet. That means they are essential in the diet. And once we take these amino acids, we get all the 20, that is the remaining 12, the body can synthesize. The net in the amino acid pool that the body has, we have these amino acids coming from the diet also, in addition to the other 12 amino acids. Totally 20 amino acids are available for the system cell to synthesize its own proteins, body proteins. Now, what are the sources that we get? Cereals are there, and pulses are there, and nuts, other things we take. But if you take the cereals, cereals contain all the amino acids except one essential amino acid that is called the lysine. So the dietary essential amino that is in low amounts, deficient amount is in the cereals is lysine. Similarly, when we take the legumes, the pulses, and that is deficient in methionine. But what is deficient in the pulses is available in the cereals. And what is deficient in the cereals is available in the pulses. That means when we make a judicious mix-up of these two in our diet, we get both the missing amino acids and totally we get the dietary essential amino acids that are required. The next point is that we have to take sufficient quantity of proteins in our diet, and then only our system will be having enough amino acids in total, and that will make up for the requirement from the amino acid pool. From the amino acid pool, the amino acid required for synthesis of various body proteins. Now, if I take some of the body proteins, insulin, for example, insulin synthesized by the beta cells and the islets of Langerhans of pancreas. Now, insulin is a composition. Insulin contains 51 amino acids in one molecule. 50 amino acids are there. Out of these 51, 17 amino acids have to be taken in the diet. They are called the dietary essential amino acids. 17, that's 1/3 of the insulin protein we have to take in our diet. If you miss even one single amino acid, that means it will cause a deficiency. So we have to take all the 17 amino acids that the insulin requires to synthesize the structure of insulin. Similarly, glucagon. Glucagon is also another small protein, peptide. And it has got 29 amino acids. Out of the 29 amino acids, 9 amino acids are essential in the diet, and these we have to take in our food so that these beta cells will not suffer for a deficiency to synthesize these two hormones, important hormones. So that is the importance of taking these dietary essential amino acids so that the cell will not suffer for its requirement. Then there are different transport proteins. The transport proteins, for example, for the transporting vitamin A, for example, from liver to the various parts of the body, there are two transport proteins. Yes, doctor. சொல்லுங்க. Audio cut ஆயிடுதா? எனக்கு இப்ப slides அந்த ஆர்டர்ல வரல, but I am continuously talking. That we have to rearrange the slides. Okay. Audio cut ஆயிடுதா? Okay.
Good evening everyone. My project title is Knowledge, Attitude and Practice Towards the Use of Vitamin C, Vitamin D and Zinc Supplements in COVID-19 among Healthcare Sectional Study, and my abstract ID is 91. Coming to the rationale, COVID-19 has already caused lots and lots of burden to the society, especially to the healthcare system, and we are at the utmost need of any of the intervention that could reduce the severity of this infection. And there are some literature evidence that suggests that vitamin C, vitamin D and zinc supplements has antiviral and antimicrobial property that could effectively improve the immune response of the host. And there are also recent studies that suggest that providing vitamin C, vitamin D and zinc supplements has reduced the severity of COVID-19 in some cases. And this study has been conducted to understand the perception of the healthcare professionals regarding the use of these supplements, that is vitamin C, vitamin D and zinc supplements in COVID-19 as an adjunctive therapy among healthcare professionals, as they are the frontline workers in this COVID-19 pandemic. Coming to the literature review, these are all the literature evidences that supports my study by implicating the importance of vitamin C, vitamin D and zinc supplements in COVID-19. The key results of these study were providing these supplements has reduced the severity of COVID-19 in some of the cases. Coming to the aim and objectives, the aim of my study is to understand the knowledge, attitude and practice of healthcare professionals regarding the use of vitamin C, vitamin D and zinc supplements in COVID-19, and the objective is to assess the KAP towards the use of vitamin C, vitamin D and zinc supplements in COVID-19 among healthcare professionals, and also to assess and provide the qualitative and quantitative information of the study. Coming to the methodology, it is a cross-sectional online survey study conducted for a period of 4 months, and the sample size was estimated to be 384. The study instrument which was used is a study questionnaire that consists of 24 questions comprising of demographic characteristics, knowledge, attitude and practice regarding the use of vitamin C, vitamin D and zinc supplements in COVID-19. And my study questionnaire is based on Math Plus protocol that has been followed in US for treating COVID-19 patients. And regarding the mark allotment, for each correct answer, the participants received 1 point, and the minimum of 6 points was considered to be adequate for each section. Regarding participant selection, the inclusion criteria for my study were all the healthcare professionals who provide concern to the study, that include medical doctors, nurses, pharmacists, biomedical experts and other healthcare professionals like physician assistant, physiotherapist, language pathologist, etc. And people who work on other aspects of COVID-19 like data scientists, artificial intelligence and machine learning were excluded from the study. Coming to the results, coming to the age group, the patients who are at the age of 21 to 35 years of age are more in number in my study, and they contribute to about 42.19% of the study population. And the mean standard deviation of the age of the participants in my study was 39.66 plus or minus 11.1. Coming to the gender distribution, male were more in number, predominantly more in number than female in my study, and they contribute to about 55.21% of the study population. Regarding the department of work, pharmacists and the medical doctors were more in number in my study, and they both contribute to about 70% of the study population, and biomedical experts were least in number in my study, and they contribute to about only 3.13% of the study population. Regarding the area of residence, the urban healthcare professionals were more in number, followed by rural healthcare professionals, and the least number of healthcare professionals were found in the suburban group in my study population. This is the table that shows the correlation of demographic characteristics with the participants' knowledge, attitude, and practice. From this table, we can see clearly that around 85% of the population had adequate knowledge, around 87% of the population had positive attitude, and around 80% of the population had followed appropriate practice. And from the age group, you can see clearly that there exists a significant difference in all the sections, that is in section of knowledge, attitude, and also in practice. The most important concern that has to be addressed immediately is that the age group of 51 to 65 years and more than 65 years healthcare professionals had possessed less knowledge when compared to the other two groups. This problem has to be addressed immediately because this age group healthcare professionals are the decision makers in most of the hospital settings. Regarding the gender, the male participants had more knowledge than female in my study, and regarding the department of work, there exists a significant difference between all the 5 professions, and it has been noted that medical doctor has more knowledge when compared to the other 4. Regarding the area of residence, urban healthcare professionals had more knowledge when compared to the other 2. Coming to the conclusion, from the study we can see clearly that most of the healthcare professionals had good knowledge, positive attitude, as well as they followed appropriate practice towards the use of vitamin C, vitamin D and zinc supplements in COVID-19. And it is very important to keep the immune system well balanced during this COVID-19 pandemic, as these micronutrients as well as these vitamins can improve the immune response of the host, that will help to combat effectively against this infection. And so I would like to conclude here by saying that lack of knowledge among the age group of above 50 years healthcare professional has to be addressed immediately, and the knowledge has to be imparted to them by any means. These are all the references of my study. Thank you.
Welcome, respected experts and my dear friends. I am a PhD scholar and I am presenting a part of my PhD study. The title of my presentation is Psychometric Properties of Activity-Specific Balance Confidence in Type 2 Diabetic Patients. Abstract ID is 1A2. 1A2. Fall is reported to be the second major cause of death worldwide. A fall is defined as an event which results in a person coming to rest inadvertently on the ground or floor or lower levels. 30 to 40% of the falls are reported each year, of which nearly 10 to 20% succumb to injury, hospitalization and/or death. Falls were outlined mainly among the participants with uncontrolled diabetes, stroke, and those using walking aids. The ignorant psychological concern with falling includes activity-specific balance confidence. Balance confidence was strongly associated with the increased risk of falling. Need of the study. With the increasing prevalence of type 2 diabetes mellitus in India, there is a need for a comprehensive instrument for assessing the balance confidence for these populations. Objective. The objective of this study was translating and determining the validity and reliability of activity-specific balance confidence scale in type 2 diabetes patients. The description about the scale. The activity-specific balance confidence scale is a self-reported measure of balance confidence in performing various activities without losing balance or experiencing a sense of unsteadiness. Activity-specific balance confidence questionnaire consists of 16 items, which includes walking around the house, walk up or down the stairs, sweep the floor, etc. Step 1. Content validation. For content validation, I met the experts in the field of diabetology, physiotherapy, and medical social work having in-depth expertise in treating as well as from research point of view in type 2 diabetes patients. For this purpose, a direction sheet, scale, and a checklist were directly given to the experts. A choice of agree or disagree, indicating an item in the scale as acceptable or unacceptable respectively. The acceptable implies that an item on the scale is acceptable and valid for these population. An opportunity to delete, keep, or rectify each item on the scale was given to the evaluators. The investigator then collects the validated scale from the expert personally. On analyzing the validated scale, the experts have recommended keeping all the items in its original form of the questionnaire for the study. Step 2 is translation. The forward and backward translation was followed as per the guidelines set by the WHO. The translators, before commencing the process, documents of a consent form, work statement definitions which were important to complete the translation process was given to them personally. The forward translation was completed by 2 qualified Kannada translators who singly translated from English to Kannada version of the scale. After translating the scale, research team and the translators reviewed the forward translation and developed the first version of the Kannada, which was handed over to an independent translator for the back translation. The second meeting was held between the translator and the research team to develop the final version of the Kannada scale. The pre-test reliability was performed on a random sample of 30 individuals participated in the pilot study. Step 3. Pre-test reliability of the Kannada and the English version of the activity-specific balance confidence scale was performed among the participants who were 30 years and above, having type 2 diabetes mellitus, good cognition, and willingness to participate. Those participants who were unable to comprehend the instruction, unfit of hearing and vision, and suffering from terminal illnesses was excluded from the study. The participants were recruited from the OPD, diabetic clinic, and laboratory services from a hospital in Karnataka, India. The participants answered the self-described scale and, if required, the questions were read out to the participants. The participants' details of the age and gender were recorded. The study was approved by the Institutional Ethical Committee and obtained the informed consent from the participants. Data analysis. The content validity of the scale was performed using the content validation index. For the purpose of the reliability analysis, Kovacs alpha statistics was used. The alpha coefficient of more than 0.70 was considered satisfactory. Results. The reliability of the scale for Kannada as well as English is 0.96 and 0.93 respectively. The overall Kovacs alpha for the English and Kannada version of the questionnaire of the activity-specific balance confidence was 0.93 and 0.96 respectively. The overall analysis of the English and Kannada version of the activity-specific balance confidence questionnaire has shown to have a good applicability to investigate the balance confidence among type 2 diabetes mellitus patients. Limitations is its small sample size. Acknowledgement. I wish to give thanks to Dr. Ramdas Naik, Professor, Department of Pathology, Yelupoya Deemed to be University, and Dr. Ibrahim M. Joshua, Associate Professor, Senior Scale Department of Physiotherapy, Kasturba Medical College, Bangalore, Manipal Academy of Higher Education. I wish to extend my thankfulness to Powell LE and Vias AM for giving the permission to use the questionnaire. I sincerely extend my gratitude to Dr. Shaini B. Sheikh, Associate Professor and Diabetologist, Yelupoya Diabetic Clinic in Charge, Yelupoya Medical College Hospital. Mr. Shivakumar M.R., Associate Professor, Yelupoya Physiotherapy College, and Dr. Irene Verghese, Assistant Professor, Department of Medical Social Work, Yelupoya Deemed to be University, for providing their knowledge as well as for validating the questionnaire. I appreciate the suggestion given by Dr. Acer George Samuel, Associate Professor, Maharishi, Markandeshwar University, Milana. I am glad to thank my participants for their kind cooperation. I am thankful to the teaching faculty and non-teaching staff of Yelupoya Deemed to be University for their support. References. Thank you.
Good afternoon to everyone. I am Renuka Sundari, pursuing PhD under Dr. G.K. Pal sir, Professor Senior Scale in Department of Physiology, JIPMA, Puducherry. I am here to present a piece of my work entitled Assessment of a Short Course Yoga Practice on Cardiometabolic Risk, Fetal-Maternal Outcomes, and Psychophysical Health in Gestational Diabetes Mellitus. According to American Diabetes Association, GDM is defined as any degree of hyperglycemia that is recognized for the first time. GDM ranges from 3.8% to 41%, and in Tamil Nadu, it is reported to be 22%. GDM is characterized by insulin resistance and it resembles type 2 diabetes mellitus. GDM has been associated with adverse health outcomes for both the mother and the newborn, and it also increases maternal cardiometabolic risk. Women with GDM are known to have decreased quality of life and increased risk of cesarean section, gestational hypertension, preeclampsia, and type 2 diabetes. Yoga is known to improve quality of life, decrease glycemic load, and reduce cardiometabolic risk in diabetes mellitus. However, the effect of yoga in gestational diabetes mellitus patients has not been studied. In GDM, there are various categories of problems. The first category is due to genetic and epigenetic modifications. There occurs inflammation, endothelial dysfunction, metabolic syndrome, and because of lifestyle changes, all these factors will lead to cardiometabolic risk in gestational diabetes mellitus. And maternal hyperglycemia. Due to glucose overload, the glucose will transfer to the fetus through placenta, and in response to the hyperglycemia in the fetus will start producing insulin, which will result in fetal hyperinsulinemia. And combination of this hyperglycemia and hyperinsulinemia in the fetus will result in macrosomia, that is increased fetal size. Macrosomia will produce many fetal and maternal complications such as prolonged labor to the mother and vaginal tear to the mother, and also fetal hypoglycemia. The third component is that the gestational diabetes mellitus women will obviously have increased stress because of poor maternal and fetal outcomes, and thereby there will be quality of life will be decreased. These are the three components, categories of problems in the gestational diabetes mellitus, and therefore I'm assessing the three categories of problems in my study. Aim of my study is to evaluate the effectiveness of yoga on cardiometabolic parameters, psychophysical health, and maternal and fetal outcomes in gestational diabetes mellitus patients at JIPMA Hospital, Puducherry. I have recruited 15 gestational diabetes mellitus women who were admitted in the obstetrics and gynecology ward of JIPMA at 35th or 36th week of gestation. And the parameters recorded were cardiometabolic parameters such as heart rate, blood pressure, and rate pressure product, and the glycemic status, and perceived stress scale, score, quality of life score were assessed, and the feto-maternal outcomes were noted. This table shows the yoga intervention which I have given to my subjects, which mainly includes few basic asanas and slow pranayamas consisting of Anulom Vilom, Chandranadi Pranayama, Brahmari Pranayama, Shritali, and finally relaxation in the left lateral posture. The total duration is approximately for 30 minutes. I have screened 54 subjects, among which 23 met inclusion and exclusion criteria. Among them, 15 consented to recruit into my study. Following the recruitment, anthropometric indices, heart rate, blood pressure, glucose levels were collected from their case sheets, and the stress level and quality of life were assessed at 35th or 36th week of gestation. Following the pre-interventional assessment, the yoga intervention was given for 10 days. Following yoga intervention, again the parameters were assessed and their feto-maternal outcomes were also noted after delivery. These are few pictures of which was taken during the yoga therapy session, which include central picture is the Tadasana and other few basic pranayamas. Table 1 shows comparison of anthropometric parameters, heart rate, blood pressure, blood glucose, stress scale, and quality of life before and after yoga intervention, among which the weight, BMI, basal heart rate, systolic blood pressure, diastolic blood pressure, and rate pressure product, which is abbreviated here as RPP, were considerably reduced following yoga intervention, which were statistically significant. Their blood glucose was also reduced following yoga intervention, and their stress level was also reduced, and there was improvement in the quality of life, which were also significant statistically. Table 2 shows feto-maternal and neonatal outcomes, which were assessed. Among 15, 10 GDM women had their normal delivery between 37 to 40 weeks of gestation, 9 had spontaneous vaginal delivery, 12 had no pregnancy complication, 11 fetus had no complications, 13 neonates were scored more than or equal to 7 at first minute in the Apgar scoring, and all the 15 scored more than or equal to 7 at the 5th minute in the Apgar scoring. And 10 among 15 neonates had their birth weight in normal range between 2.5 to 3.4 kilogram. 10 days of practice of yoga, consisting mainly of pranayamas and basic asanas, resulted in decrease in weight, BMI, heart rate, SBP, DBP, and rate pressure product, which is abbreviated as RPP. This rate pressure product is a myocardial stress, work stress, and it is an index of cardiovascular risk. And their blood glucose was considerably reduced. Their stress level was reduced and quality of life was improved, and there was also improvement in the feto-maternal outcomes as well. This is the first study in the world to report that 10 days of yoga practice in their 3rd trimester of pregnancy had improvement in the feto-maternal outcomes. These are my references. Thank you.
Respected Chairperson and distinguished delegate, I am Dr. Indranil Manna, Assistant Professor, Department of Physiology, Midnapur College Autonomous, Midnapur. I am very much pleased to being invited for the International Conference and 41st Annual Conference of Indian Association of Biomedical Scientists. Today, I am going to deliver my topic, Studies of Anthropometric Physical Fitness and Physiological Variables of Tribal and Non-Tribal Female Children of 12 to 18 years. As you know, that sports talent may be identified among the children at very early age when they show interest in different sports. Anthropometric physical fitness and physiological variables contribute to selection procedure in different sports events. Women participation in sports is increasing at present time, and many women athletes getting success in national and international area. On the basis of the above, the present study has been designed to investigate the anthropometric physical fitness and physiological variables of tribal and non-tribal female children of 12 to 18 years. For this study, we have selected 600 female children between 12 to 18 years, and they were selected from after proper medical checkup from the West Midnapur district, West Bengal, India. And they were equally divided into two groups, tribal and non-tribal, and having 300 subjects in each group. These groups are again subdivided into 7 subgroups. Okay, so similarly, they have 12 years, 13 years, 14 years, 15 years, 16 years, 17 years, and 18 years age group. And each age group have 50 subjects. The subjects were informed about the possible complication of the study and gave their consent. The institutional ethical committee approval was also taken for the study. The anthropometric variables, including measurement of height, body mass, skin fold thickness, were taken for assessment of body fat, estimation of waist-hip ratio, measurement of mid-upper arm circumference were taken. The physical fitness variables, including elastic leg muscle strength, abdominal muscle strength, grip muscle strength, measurement of anaerobic power, measurement of speed, flexibility were performed. The measurement of physiological variables, including blood pressure, heart rate, and pulmonary functions were performed. The VO2 max was assessed by Queen's College step test. All the variables of anthropometric physical fitness and physiological variables were expressed as mean and standard deviation. An ANOVA followed by multiple comparison test was performed to find out the significant difference in selected anthropometric physical fitness and physiological variables among the groups. The result shows a significantly lower body mass and body surface area was observed among the tribal children when compared to non-tribal children of some age group. In addition, a significantly increase in height, body mass, BMI, and BSA were observed with the advancement of age of the children. The possible reason for the increase in height, body mass of the children might be due to the osteoporotic response to exercise. The osteoporotic effect of exercise is depend on load dynamics of the volume, intensity, and duration of the training administered to the individual and the period of life when exposure occurs. We have also measured the body fat, waist-hip ratio, and mid-upper arm circumference, and it has been noted that there was a significantly lower fat mass and waist-hip ratio was observed among the tribal children compared to non-tribal children of some age group. Further, significantly lower percentage of body fat, fat mass, higher lean body mass, waist-hip ratio, and mid-upper arm circumference was observed with the advancement of age of the children. The percentage of body fat plays an important role for the assessment of physical fitness of the player. The waist-hip ratio has been shown to be related to the risk of coronary heart disease, and the mid-upper arm circumference is a measure of nutritional status. So these variables were measured, and these changes might be due to the level of maturation factors and or motivation and exposure to long-term and high-intensity training among the children. The physical fitness variables were measured, and it has been noted that there was a significantly higher flexibility, speed, and power was noted among the tribal children when compared to non-tribal children. Further, significant increase in flexibility, speed, power were observed with the advancement of age in both tribal and non-tribal children. And these changes might be because of the level of maturation factors and or motivation and exposure to long-term and high-intensity training among the children. The significantly higher strength of back, grip, abdomen, and leg muscles were noted among the tribal children when compared to non-tribal children in some age group. Further, significant increase in strength of back, grip, abdomen, and leg muscles were observed with the advancement of age in both non-tribal and tribal children. These changes might be because of the level of maturation factors and or motivation and exposure to long-term and high-intensity training among the children. We have also measured the VO2 max and hemoglobin level among the children, and it has been seen that there was a significantly higher VO2 max among the tribal children. However, there was no significant difference was noted among the in hemoglobin level among the children of tribal and non-tribal age groups. The increase in VO2 max may be due to increase in arteriovenous oxygen difference and stroke volume. Moreover, these changes might be due to because of increased volume of endurance training. We have also measured the lung volume and capacities, and the result shows there was a significantly higher FVC, FEV1, and peak expiratory flow rate was observed among the tribal children when compared to non-tribal children of some age group. Further, significantly higher FVC, FEV1, and peak expiratory flow rate was observed among the tribal and non-tribal children with the advancement of age. And these values shows that the changes might be due to the maturation level and or exposure to high level of training among the children. It can be concluded that identification of children at early stage of their growth and development may produce elite athlete in future. Talent identification also can be used as a counseling technique that helps to discover and explore area of talent for particular athletes. In order to reach their goals, young children should be subject to a series of tests reflecting body composition and cardiorespiratory fitness profile, which will indicate their present overall strength and weakness. It has been observed that the tribal children has a better body composition and cardiorespiratory fitness variable than the non-tribal children. It can be suggested that more emphasis should be given on the tribal children for identification of potentiality and talent in sports. So these are the references we have used for the present study. Now the author we acknowledge RUSA 2.0 Component 8, New Delhi, Government of India for financial assistance to carry out the research work. The author sincerely and wholeheartedly acknowledge the contribution of children participated in this present study. The author are also thankful to the UG and PG students and the coaches and the laboratory staffs for extending their support for the present study. Thank you, all of you.
Good morning, all. Today's topic is lived experience of women with ischemic heart disease, a pilot study on phenomenology. It's a qualitative study. The objective of the study are explore the lived experience of women with ischemic heart disease. The research questions are, what are the experience of women with ischemic heart disease? What are the lifestyle changes taking place in women after ischemic heart disease? What are the challenges of women living with ischemic heart disease? What are the factors that facilitated the healing process? And what are the factors that hindered the healing process? The methodology of the study, it is a qualitative study where the phenomenological approach is used. Here, the in-depth interviews are done from the patient. Setting of the study is Malabar Medical College Hospital, Calicut. 2 sessions of data collection has taken place. That is first one from cardiology outpatient department and second round from their home city. And the population is 10 women with ischemic heart disease. This interview is lasting for 45 to 60 minutes and the 2 interviews were done per participant. The data collection before that, informed consent was taken from the participant. The researcher itself conducted the interview and the interviews were audio taped and it is an unstructured open-ended interview. The questions, examples of questions like, tell me about your experience of affected with heart disease, tell me about your experience after arriving home from the hospital, etc. And the data analysis by using collaces method of data analysis. Here the tapes are transcribed and this transcribed verbatim is being read and reread in order to obtain a general sense about the content. And from this transcription, significant statements were extracted and the meanings were formulated. And these formulated meanings, which is called codes, otherwise, were sorted into themes and sub-themes by using a software called NVO, which is a software version 9 is being used. And the findings were integrated into an expositive description. And the validation of the findings also will be done by the participants by giving the transcripts to the participants. And concurrent coding and analysis being carried out. Coming to the significant statements from the transcribed verbatim, it is the statements that are of direct relevance to the phenomenon under investigation. That is the significant statement. And this is extracted by reading and rereading the transcribed verbatim. For example, here we can see Mrs. A said, it started, that is the pain started early morning at 4 o'clock. I went to bathroom, came back and lay down. When I was stretching, I felt like some pain from here, that is showing the chest, like something obstructing. So this is said by the participant 1 in their transcription, page number 2 and line number 22, where it is being described. So that is how from the transcribed verbatim, significant statement is being taken out. Then the formulated meaning. From here, that is from the significant statement, how the formulated meaning is being taken out. So here, again we can see the example. That day when the pain started, I had done lots of work, and just before that, I go for cleaning the courtyard of nearby house, where I used to go for household work. So here, from this significant statement, it can be seen that it is observed that the onset of pain was mostly while doing some activities. So that is the formulated meaning which is being taken or which is being used. Then from such significant statements, we have to derive the sub-themes. So here again we can see participant 1 and 2, participant 1, 3 saying about the description of the pain. Pain started in the early morning, 4 o'clock. I went to bathroom, came back and lay down. When I was stretching, I felt like some pain from chest, something obstructing. So such significant statements clustered together to make a sub-theme of agonizing pain. So similarly, such sub-themes are together made to put themes. Like here, agonizing pain and some other symptoms which is expressed by the patient, that is being put as a fluctuating symptoms to make a theme of perception of pain. Similarly, that fear of impending death, you can see anxiety of theme is made by irrational reasoning, anxiety about future of loved one, being burden to the family. Similarly, we made a theme anxiety. Similarly, themes like stumbling block, support systems, learning to cope with various ways is being taken out. So these are the sub-themes and themes. And from these themes and sub-themes, it is being described, like for perception of the pain, that is the theme. This theme is described the physical distresses expressed by the participants. So here, as I have already shown, agonizing pain, fluctuating symptoms, the description of the patient is being taken out. Like so here, similarly, we can see when I reached the hospital, there was full of sweat, and also I had strangulating pain. The pain was like something kept on my chest. So that is a description given by the patient regarding the sub-theme of agonizing pain. Similarly, fluctuating symptoms, fear of impending death. Here we can see the themes reveal the fear of death embedded in the patient. So here you can see the patient's description. After knowing this, that is the disease condition, I thought I may die. Anyway, my grandfather and all died due to this only. After the third attack, he had gone. So similarly, we can see the expression of the patient. So that is the fear of impending death. To make a theme of that, we have given the description of the patient. Similarly, anxiety, where sub-themes you can see anxiety about the future of loved one, fear of being burdened to the family, etc. Again, the fourth theme, stumbling blocks, where you can see the sub-theme of financial constraints, mobility restrictions, dietary disturbance, and the theme of support system, you can see the sub-theme of family support, social support, and spiritual support, and the theme of learning to cope various ways. Here we can see this theme try to describe the various coping strategies adopted by the participants. So here description of the participants also be given. So similarly, the themes and sub-themes together to make a phenomenon. That is a description of the phenomenon of lived experience of human with ischemic heart disease. So from out of all these themes and sub-themes, the phenomenon is emerged out. That is made conceptual model. That theme, that phenomenon is that to live is to suffer, to survive is to find some meaning in the suffering. So here in this model, we can see all these themes, sub-theme together. We can see when we infer or we can see that the phenomenon that is to live is to suffer, to survive is to find some meaning in the suffering is being displayed. So here, the study revealed the lived experience of women with ischemic heart disease as a complex, life-changing actuality which involving the dynamic interplay between physical, psychological, social, spiritual, and economic dimensions based upon phenomenology. So this is the study. Any questions you can ask?
one and all. I am Urvashi, abstract ID 197, and today I am presenting on homology modeling, screening, and MD simulation of selected compound from public database on DBL3X domain of Plasmodium falciparum erythrocyte membrane protein 1 involved in placental malaria. So, malaria in pregnancy caused due to gathering of parasite-infected erythrocyte in the placenta, in which Plasmodium falciparum erythrocyte membrane protein 1 interact with chondroitin sulfate on the placental surface. Clinical complication of infection during pregnancy seems to be high, like low birth weight, vaginal bleeding, abortion, premature delivery, hepatitis, and jaundice. So the main objective was to characterize different properties of protein, Plasmodium falciparum erythrocyte membrane protein 1 in silico, like identification and mining of appropriate receptor protein, and also the ligand molecule which can easily bind with it from different public databases. So this is a workflow of project in which we can for target discovery we did a literature review from different public databases and find a pathway in which different proteins we get, from which PFEMP1 is mainly involved in a pregnancy related malaria. So we took that and characterized the different properties of that protein. Then a domain of particular protein we model using a modeler software, and model protein further simulated in a GROMACS with 10 nanosecond for checking of stability, checking stability of a protein, and then model protein using PROSA server and PDB-sum analysis. We check the quality of model protein, which shows it has a good quality. Then screen it against different ligand molecules from public databases, and the best one is further docked in FLAC-X. Then the complex of protein and ligand is simulated in GROMACS again for 1 nanosecond, and the result is analyzed with different graphs. So active site of DBL3X domain. So in here image structure shows that active site present in yellow color binds to sulfate binding loop, and sequence showing residues involved in a sulfate binding loop which is conserved side chain. So here from 109 to 119 are residues which binds to CSA, which we can target using different ligands. So in a domain DBL3X, total 360 residues are there, which shows break in turn region. So PDB database has protein with break. So we model that protein structure successfully using modeler software and created a model without break. So further created model is validated using PROSA analysis, which is available online, and the quality of model protein is checked, which is a good. And also the Ramachandran plot was created, which shows the most of the amino acid are in a allowed region, and so the our created protein structure is a which is has a good quality. So after creation of a successful model of protein, molecular dynamic study using some basic law of physics is done using a GROMACS software, in which we can see at particular pressure, temperature, and density, protein shows a stable structure. So in first graph, we can see that protein is converging after 1300 steps and showing stability. And at below, there is a structure of protein before and after simulation, which we can see there are some loops get compacted and structure is more stable. After simulation, different drug molecule is taken from databases like Drug Bank, Campbell, malaria data, and perform a virtual screening from which we get some good results, which is bind to the domain of DBL3X and interact with the active residues. So the molecule with the good binding capacity is taken and ligand with ID Campbell 1387961 proved to be the best compound to be docked against DBL3X domain receptor, showing the lowest binding energy of -18.77, which shows the stable structure of interactions, for which FlexX software is used for the docking process. After successful docking of ligand and protein receptor, again the simulation of the complex is done in GROMACS with 1 nanosecond, which shows the structure of complex, and complex is very stable at particular pressure, density and temperature. And also the RMSD plot is drawn, which shows not more fluctuations. So the ligand molecule is very very stable, and we can use it as a drug molecule for a further study. From the image we can see that after simulation also the complex structure of protein and ligand not showing much deviation, and structural conformation is same as it earlier, and there is no loss of functions or break in the structure, suggesting that the complex is well stable after 1 nanosecond simulation. So we can conclude that in silico approaches have proved that computational tools like homology modeling, virtual screening, simulation, docking can aid in designing effective vaccine or drug candidate within shorter span, and it need to be further tested in wet lab for verifications, and we can do a trials on that. Thank you.
Good day to everyone. Myself, Dr. Tugalbai Tagayev. I'm an assistant professor at the Department of Hospital and Internal Medicine at Kyrgyz State Medical Academy here in Bishkek, Kyrgyzstan. My specialty is cardiology and rheumatology. So hopefully you're going to enjoy this research work. This research work we have done with another colleague, and it's related to my specialty. So briefly, this research work is about anticoagulation of elderly patients with certain diseases like atrial fibrillation. So let's remind ourselves, what is anticoagulant? So anticoagulants are commonly known as blood thinners, are chemical substances that prevent or reduce coagulation of blood, thus prolonging the clotting time. So we use anticoagulants to prevent secondary complications in the setting of certain diseases, like atrial fibrillation. So some of the patients, they're prone to develop some thromboembolic complications in atrial fibrillation, like hemorrhagic stroke. So this is the retrospective study where we studied the charts or cards of the patients and we examined their adherence to taking this anticoagulants. So if we're going to use anticoagulants on a chronic basis, they're going to reduce the risk of stroke by 64%, and they're going to reduce the risk of death by 25%. So it's important that if we're going to use the anticoagulants on a chronic basis, the development of stroke, specifically hemorrhagic stroke, decreases by 64%. The present study is aimed to assess the treatment adherence in elderly patients with non-valvular atrial fibrillation who were prescribed the vitamin K antagonist, warfarin. As we know that there are different types of anticoagulants, and one of the type of anticoagulant is vitamin K antagonist that inhibits the synthesis of certain clotting factors in liver. That's warfarin. Materials and methods. In the present retrospective study, we analyzed the medical records of 202 elderly outpatients patients with non-valvular atrial fibrillation aged between 65 and 74 years. Atrial fibrillation, so it's one of the most common types of cardiac arrhythmias. Among other, there are different types of cardiac arrhythmias, as you know. So affecting roughly 3% of the population and increasing with frequency with age. So there's a positive correlation. When the age increases, the rate of the atrial fibrillation also increases. As you can see from this diagram, according to different literature, it's around 6% if the patients are from 60 to 69, and it's around 16% if the patient's age are between 70 and 89, and it's around 22% if the patient's age is above 90 years old. Mortality. So prevention of stroke, specifically hemorrhagic stroke in patients with atrial fibrillation, remains a particularly important public health problem around the globe, and as 70 to 80% of patients die or develop disabilities after suffering a stroke. So most of the patients, they die, but if they suffer, there will be long-term disability problems. Patients, so now there are certain moments that you have to know. Patients were excluded if their charts contained no information about treatment adherence. In all patients taking warfarin, we determined how long the international normalized ratio remained in the therapeutic window. So that's about the international normalized ratio. All patients had a high risk of thromboembolic complications on the CHA-2D S2V scale, and a low risk of hemorrhagic complications on HAS-BLED scale. So therefore, all patients had been correctly prescribed warfarin. Among patients with poor atrial fibrillation monitoring, we conducted a survey to determine the reasons for their irregular doctor visits and poor INR monitoring. So in addition, we devised a questionnaire to determine doctors' awareness of when antithrombotic therapy should be prescribed and their knowledge about these scales. We then delivered this questionnaire to 57 doctors from family medicine centers. So here is the diversity of the work. So there is more women involved in this study compared to men. Even this study was random. City residents and rural residents, as you can see that roughly around 50% were city residents, urban residents, and 50% around 50% rural residents in this country. And there are certain diseases or comorbid conditions that the patient have had. The most common one, I think, was the arterial hypertension, coronary heart disease, with or without history of the myocardial infarction, congestive heart failure, diabetes mellitus, type 1 and type 2, and history of acute cerebrovascular accident and chronic kidney disease. Problems associated with warfarin arose throughout the follow-up period. After 1 month of treatment, the number of patients taking warfarin had decreased to 71.3% of all patients, and less than half of the patients were still taking the drug. So as you can see that the number of people, initially it was 100%, so it decreased by 71.3%. And after 12 months, it was around 9%, 9.4%. In subsequent periods, the number continued to decrease. Of all patients who had been prescribed warfarin with periodic INR control, only 19 patients remained. So it's around 9.4% remained after 1 year. This is the real number of patients who were regularly observed during the year and followed the recommendations of the doctors at the Family Medicine Center. So only 19 remain after 1 year out of 202. So as you can see that these are the people who didn't, this is the ratio of the different factors with different conditions that didn't adhere to the warfarin treatment. So 48 people, they were rural residents, and 22, they were urban residents. So more rural residents, they didn't adhere to the treatment. The main reason in this category was there was no facility or availability to measure the international normalized ratio. So in the survey, in the questionnaire, the main reason was that they were not able to control the INR. So they were aware of the hemorrhagic complications. So this category, they stopped the treatment. Second category was medical contraindications. Several such contraindications like PUD, peptic ulcer disease, history of some colonic ulcer diseases, certain category of diseases which prevented patients from taking warfarin. So it's pure medical contraindications. The third category is outpatient doctors. So here, the doctors, they were afraid that they were misinformed or they were less informed that the patients may develop some hemorrhagic complications. So 39 patients out of 202 patients, they were not basically prescribed warfarin by outpatient doctors. And 7 patients, they were not able to take warfarin due to their chronic alcohol abuse, and 17 patients, they developed real hemorrhagic syndrome, and 33 patients, they independently switched to taking aspirin-based drugs. So I just want to highlight that 33 patients, they didn't adhere to the doctor's prescription and they independently switched to taking aspirin-based drugs. Conclusion, the results of the present study showed that anticoagulants were underprescribed at the outpatient stage in centers of family medicine in our country. In Kyrgyzstan, the main drug of choice for specialists remains warfarin, which only provides adequate therapy in a small number of patients. So it's around 9.4, or I would say that it's around 19 patients out of 202 patients. A questionnaire administered to doctors demonstrated that low awareness of the need for anticoagulant therapy in patients with atrial fibrillation with low adherence to the recommendations. It should always be remembered that the good adherence to anticoagulant therapy is crucial to the effective prevention of thromboembolic complications in atrial fibrillation. So, dear listeners, I hope you enjoyed this study. So basically, this study is about the importance of anticoagulation of elderly patients, as there's a positive correlation between the age of the patients and the frequency of arrhythmias, specifically atrial fibrillation. As the age increases, the incidence of atrial fibrillation is going to prevalence also going to increase, thus predisposing patients to develop the thrombus formation, embolus formation, and for a secondary to develop secondary complications of this disease. And one of them is hemorrhagic infarction. Thank you.
My greetings to everyone. I am Ajit Kumar, Molecular Genetics Lab, Department of Bioinformatics, Warda University. My research topic is about individual cellular release of virions by blocking the interaction between SARS-CoV-2 virus RNA and BSD2, a virtual screening approach. Introduction. We are all now on ongoing outbreak phenomena caused by novel coronavirus, which is declared as severe acute respiratory syndrome by World Health Organization. Every day, the confirmed cases and death due to this viral infection is increases. Many researchers and scientists believe human is the final ghost of this deadly virus. One of the most powerful strategies to control this spreading is genomic level surveillance. Genome of SARS-CoV. This virus belongs to Coronaviridae family, which is pseudotype with retrovirus. The coronavirus genome is longest known genome among RNA viruses, ranging from 26 kb to 32 kb in length. Open reading 7a. We are taking ORF7a for this research work because accessory gene of ORF7a proteins have been found to be important for the addition of host-mediated immunity. ORF7a has 15 amino acid N-terminal signal peptide and 81 amino acid luminal domain and 21 amino acid transmembrane domain and 5 amino acid cytoplasmic type. The previous studies refer that BSD2 does restrict SARS-CoV, but the loss of ORF7a leads to much greater restriction, confirming the role of ORF7a as an inhibitor of BSD2. Bone marrow stromal antigen 2. The BSD2 initially identified to be a pre-cell growth promoter, but it also inhibits the release of virions of the retrovirus. The interaction of ORF7a and BSD2 results in inhibition of BSD2 proliferation, which leading to reduce the tethering function of BSD2. Results. The overall workflow based on the computational approach. The SARS-CoV and SARS-CoV-2 have similar amino acid composition and protein structure. Initially, the structure and sequence element was done between them. The two just confirmed the similarity and conserved residue between the two structures. The results 85.2% is sequence identity and similarity is 90.2%. Hence, we concentrate only SARS-CoV-2 ORF7a for further process. The picture shows that structure element of SARS-CoV ORF7a and SARS-CoV ORF7a. Virtual screening and IFD docking. The overall workflow is done using Scarnet Mastery software. The screening of Cas-handled compounds and zinc database compound again SARS-CoV-2 ORF7a was finished in Bright. The top screening compounds again logged by induced method. Based on the glide score, IFD score primarily, the best infecting compounds are shown in table. Through the data set, the compound of pyridine 5-amino 2-deoxy bind with glide score of -8.083, as well as zinc FDA approved compound decitabine binds with glide score of -6.852. We can see more number of interactions through this picture. The first picture shows that interaction of ORF7a with pyridine 5-amino 2-deoxy, and second picture shows that interaction of ORF7a with decitabine. Coming to conclusion, according to the possibility of this interaction, it is concluded that the peripheral binding of predicted compounds to SARS-CoV-2, it may inhibit the interaction of host BSD2. Followed by this work, these compounds will be used for molecular dynamics studies, also for in vitro immunoanalysis to inhibit the virus and interaction with BSD2. Thank you.
Hey, hi everyone. My name is Shubham Ashok Vaitsal and I have completed my graduation from Dr. D.Y. Patil College as Biotech and Bioinformatics, and currently I am working in IQVIA Quintiles as a clinical process coordinator. And here I'm present you one PPT on the topic of molecular modeling and enrichment analysis of drug cabozantinib and drug targets of advanced renal cell carcinoma. So as we know, we heard what is renal cell carcinoma and what is carcinoma. So carcinoma is a cancer. It's caused due to abnormal growth of the cells in the body. It is also characterized as a genetic disease. And we have also working on the tyrosine kinases, and tyrosine kinases are the major family of proteins frequently dysregulated in various cancers. Kinases play overlapping and intricate roles of cell transformation and tumor initiation, survival, and proliferation. So here we have different pathways, and we are working in drug molecular modeling. So drug which we are using here is cabozantinib. So cabozantinib is a new multi-targeted tyrosine kinase inhibitor, which is TKI, that inhibits the tyrosine kinase activity of vascular endothermal growth factor receptors, which is VEGFR. And cabozantinib is a small molecular drug approved by US in 2012 for several antigenetic agents for use in renal cell carcinoma. It suppresses metastatic angiogenesis and oncogenesis by inhibiting receptor tyrosine kinases, and it is sold under the name of Carbometrics. It is available in the market with three formulations, which is 60 mg, 40 mg, and 20 mg. So in this, we have made one diagrammatic wave of steps. So in this, we have done identification of the drug, then the identification of target proteins, protein structure determinations, structural analysis, structural refinement, drug target, drug ligand preparation, ligand selection and its optimization, protein ligand docking, binding affinity, complex formation, toxicity, and enrichment analysis. So total we have done 12 steps, and we have showed in here in diagrammatical view. So these are the results we got it from DS Visualizer software, and in this we are we have shown the hydrogen bond and pi bond. So here in the green, dark green color, we can see the H bonds, which is hydrogen bonds, and in the complex file with drug and protein structure. We have used a drug as a ligand, and we have compared ligand protein and ligand drug, protein drug. So in the pink, we can see pi bonds, and we are visualizing those H bonds and pi bonds, and we are comparing it to the standard one with the drug one. In the next, we have total done almost for 13 families. In that, we have got the best results for 9 protein families. So there are different protein families like FGFR1, FGFR2, and VGFR1, VGFR2. So we have got different PDB structure IDs from protein data bank, and for that particular protein data banks, we have got best common interaction with the drug compared to the indigenous inhibitor. So we can see here there are most common interactions are 3, and there are many of the PDB IDs getting the best results here, and we have also checked with the indigenous inhibitor. So here we have checked the structure and bioavailability of the target. In this, we have checked the lipocity, physiochemical properties, water solubility, phytochemicals, phytokinetics, and drug likeness. We have checked, which is very important, and molecular chemistry. What is the chemistry of that molecule? So we have checked with all those best result protein drug molecules. The next slide, we have also checked the results for drug likeness and toxicity. So in this, we have checked the Lipinski's spiral and also the toxicity according to the substrate and inhibitors of the particular molecules. So here we have given the names of the PDB IDs. From that, we have get the best structural and properties for different Lipinski's filter analysis. So here we have checked the PDB IDs, protein molecular weight, H bond, acceptor H bond donors we have checked, and also the toxicity for the inhibitors and substrates, and also the absorption of the drug in the molecule. So in the enrichment analysis, we have checked with different disease banks like Omen disease, Jensen disease, design disease and leak. So here in Omnium we have got the results for FGFR1, MET and KIT. In the Jensen we have got the best results for breast cancer, skin cancer, melanoma cancer, etc. So here there we have also checked the p-value with all those PDP IDs, and then we have compared with.
Greetings of the day. I'm M. Janarajani from Molecular Genomics Laboratory. I'm pursuing my PhD degree under Dr. V. Hemamalini from the Department of Bioinformatics, Bharathiar University, Coimbatore. Today, I'm here to present my work, Design of Novel Peptides with Quorum Quenching Ability to Inhibit the Vibrio cholerae Progression. So, the introduction. The Vibrio cholerae, a gram-negative bacteria, which is the causative organism of cholera, it survives in the human host through forming biofilm, which is mediated by quorum sensing. Quorum sensing is the signaling between the bacterial cells based on the cell density. So at low cell density, the extracellular membrane protein CQSS and LuxPQ activates the protein LuxU. The LuxU protein further phosphorylates the LuxO protein, which mediates the production of small RNAs QRR1, 2, 3, and 4. It mediates the production of protein APHA, which plays a major role in virulence gene and the biofilm formation. And at high cell density, the autoinducer molecules CAI1 and AA2, it inhibits the production of APHA protein and induces the production of HAPR. The HAPR protein helps in the aggregation of the bacterial cells. So the interaction of LuxU and LuxO, it plays a major role in the biofilm formation. And peptides are unique classes of pharmaceutical compounds that lies between small molecules and proteins. The size ranges from 7 amino acids and 50 amino acids. So this is highly used against protein-protein interaction, as these peptides bind to the receptor protein and it blocks the interaction of other proteins. Because of this inhibiting potent, these peptides are used as drugs in the diseases like cancer, hypertension, CVD, and etc. So my major objective is to design a peptide which can inhibit the interaction of LuxU and LuxO that would reduce the biofilm formation, which can prevent the further spread of this infection. Materials and methods. It is in complete in silico method. So I have used online servers and softwares for the modeling of these peptides. The proteins LuxU and LuxO doesn't have any crystallized structure. So I went for the homology modeling. The modeling of proteins were done in GlideTask MassPro in the version 11.8. So after modeling the proteins, it were refined using 3D Refiner and was validated in Galaxy Web Server. So after the validation, the top two structures from the proteins LuxU and LuxO were subjected for protein-protein docking. The protein-protein docking was carried in Haddock server. So the 4 complexes were taken from this protein-protein docking. The template was retrieved from the complex which are high hydrogen bond. So the hydrogen bonds were viewed through this LickPlot software. We can view the interaction of proteins and the hydrogen bond between the residues of both the proteins. So the template were retrieved using LickPlot plus service. So after retrieving the template, I designed the peptides using PepFold server. So after designing the PepFold, it was subjected to protein peptide docking in Haddock server. So this is the result of my study. I've taken the first complex which have 18 hydrogen bonds and it includes 13 active site residues from both the proteins. The table shows the active site residues which is involved in the interaction of both the proteins, that is LuxU and LuxO. Here the LuxO acts as the receptor and LuxU was taken as the template. So the template sequence which is taken from LuxU is Q M R E K Q W R D A K K A E E M E G M D. So this is the result of the top the peptide protein docking. So top 3 peptides from the peptide protein docking was taken. It is based on the number of hydrogen bonds and the Haddock score. So from the design peptides, the peptides 1, 4, and 9, it shows high binding affinity, that is with high hydrogen bonds and high score. Those three were retrieved for the studies. So the first peptide with the sequence of QM, KE, KQ, WH, DA, KR, AE, NM, EG, MD with the Hardoff score of minus 99.4 and 10 hydrogen bonds. It places on the top of the table and it includes 5 active site residues, and other two peptides are peptide 4 and 9, and the sequence and the Hardoff score are minus 97.6 for 4 and minus 93.7 for peptide 9 with 10 and 9 bonds respectively. It also includes 4 and 5 active site residues in the receptor site, and this is the structure of my peptides. Peptide 1, this is the structure of peptide 4 and it is the structure of peptide 9. So from this work, I conclude that the top 3 peptides which have high binding affinity towards the LuxO protein can interact with LuxO and can inhibit the interaction of this LuxO and LuxU, and it is seen through this in silico approach. So our future perspective is to find out the stability of this 3 peptides by molecular dynamic simulations, and after the checking the stability of these 3 peptides, it will be synthesized and examined for its quorum quenching ability through in vitro studies. Thank you.
Greetings to one and all present here. I am Dr. M. Kavita, student of Dr. E. Padmini ma'am. I am here to present a part of my PhD work entitled In Silico Docking Analysis of Bioactive Compounds from Aqueous Extract of Philanthus acidus Keel's Fruits against MAPK1. Protein kinases are intimately integrated in different signal transduction pathways for the regulation of cardiac function in both health and disease. In the heart, these signal transducing proteins are engaged in the regulation of cardiac function, cation transport, myocardial metabolism, gene expression, contractile activity, cellular growth, and cell apoptosis. In view of the fact that most of these kinases are activated in heart disease and their inhibition has been shown to improve cardiac function, it is suggested that these kinases form excellent targets for drug development for therapy of heart disease. There are different types of protein kinases that we know so far. Protein kinase A, protein kinase C, abbreviated as PKC. Apart from that, we have calcium calmadiol independent protein kinase, as well as phosphoinositide 3 kinase, which is abbreviated as PI3K. And the fifth one is mitogen activated protein kinase, which is abbreviated as MAPK. Under MAPK, you have subdivisions like extracellular signal regulated kinase, which is abbreviated as ERK1 or 2, C-Jun N-terminal kinase, JNK1 and 1 to 3, P38 MAPK, and ERK5. MAPK is a family of serine threonine specific protein kinases. There are 4 distinct subfamilies of MAPK, as just now we have seen: ERK1 and 2, JNK1 and 2, 3, P38 MAPK, and ERK5. MAPK1, or which is otherwise ERK2, is 41 kDa protein consisting of 360 amino acids. Subcellular location of MAPK1 or ERK2 protein is in the cytoplasm and the nucleus. Upon activation by dual phosphorylation on its tyrosine and threonine residues by upstream kinases, MAPK1 is translocated into the nucleus from cytoplasm, where it phosphorylates its nuclear targets. The ERK pathway has been found to be responsible for the phosphorylation of BCL2 that contributes to cell survival, the suppression of the apoptotic effect of BAD, the upregulation of the anti-apoptotic protein MCL1. Moreover, it has been also shown that ERK1 and 2 is one of the regulators of TP53 protein accumulation and activation during the DNA damage response. So here we can see a picture showing your how the cardiac cell growth is promoted by extracellular signal regulated kinases 1, 2, and the apoptosis and cell death is actually regulated by P38 kinase. Now, the plant under study is Phyllanthus acidus, a fruit, which Phyllanthus acidus is commonly called as star gooseberries in English and Aranelli in Tamil. It belongs to the family Phyllanthacea. It is a plant bearing edible small yellow berries fruit belonging to the family Phyllanthacea. It is commonly grown in Indonesia, Malaysia, Vietnam, and in Indian home gardens. People eat gooseberries due to its pungent, sour, and astringent taste. They are used as blood purifier, liver tonic, and as appetite stimulant. Hence, the aim of the present study is to identify the bioactive compounds in the aqueous extract from the fruit of Phyllanthus acidus by LCMS and in silico docking analysis of bioactive compounds from Phyllanthus acidus aqueous extract of fruits against MAPK1. So the materials and method involved were collection and identification of the plant material, number 2, preparation of plant extract, number 3, LCMS analysis, and number 4, in silico docking studies using the various tools. The results, coming to the LCMS spectrum of the Phyllanthus acidus aqueous fruit extract which is given, also the compounds that is isolated, some of the important compound which having important pharmacological properties are given on the right hand side of the slide. So the docking studies were performed. The first step towards the docking studies was to retrieve the structure and the sequence of MAPK1. A UniProt database was used to retrieve the sequence of MAPK1, as well as the PDB database was used to derive the crystal structure of ERK2 or MAPK1. So here is the crystal structure of MAPK1. Then for further docking analysis, certain bioactive compounds from the Phyllanthus acidus aqueous fruit extract was selected, namely methylprednisolone, tridesacetokivorin, hydroxysalmeteriol, dexatetraenoic acid, and trihomovitamin D3. These were the 5 compounds which were selected based on the biological function. The 2D structures of these bioactive compounds were generated using ACD ChemSketch tool. Open barbell converter tool is used to convert this file into the PDB format, and that is used during docking analysis. Further, AutoDock tools were used to analyze docking simulation. So based on the lowest energy, the best ligand receptor structure was found. So here are the results where the interaction between MAPK1 and trihomovitamin D3 is shown. This figure shows the interaction between MAPK1 and hexadecatetraenoic acid. This third picture shows the interaction between MAPK1 and methylprednisolone. This is with the fourth compound, interaction of the MAPK1 with hydroxysalmeterol. And this is MAPK1 and tridesacetokivorin. This is the overall docking results. And from the table, it is clear that the best ligand receptor structure is found to be that of tridesacetoxykivorin, because the number of hydrogen bonds formed is the maximum, namely 5, and the least energy is involved, namely minus 7.79. Thus, it is concluded that bioactive compounds from fruits of Phyllanthus acidus plant may have cardioprotective potential. However, further wet lab studies have to be performed to confirm it. Thank you.
good morning to you all. I am Dr. Srinivasan Jayaraman from University Tunku Abdul Rahman, Malaysia. I am happy to be a speaker at the International Conference on Recent Trends in Biomedical Research, organized by Indian Association of Biomedical Scientists. Today I will be talking about the potential of zinc oxide nanoparticles to be a right choice for antibiotic-resistant pathogens. We know well that nanotechnology is a fast-growing technology. We can see in human population and also increase in microbial infections. Need more medication therapy, which are insufficient currently. And also the development of antibiotic resistance is another problem to treating the microbial infection. So now, to overcome all these issues, that means the increase in the microbial infections and also demand for available medications, and most importantly, the antibiotic resistance against the microbial infections. All these demand for all these factors, a demand for a new and compelling technology which can provide a promising solution to completely treat the microbial infections. So we know antibiotic resistance were restricted to the hospital environment earlier. Now it is there everywhere. Because of globalization and excessive use of antibiotics in animal husbandry, and specifically the use of multiple broad-spectrum antibiotics, are responsible for the spread of antibiotic resistance, mostly. So we can see that in the year 2050, there will be no effective antibiotic available if no new drug is developed or discovered. So that's a problem now. That is the critical issue now. So that's why there is a critical need to search for alternative method of controlling antibiotic resistant pathogens. So we can see from here, there is a critical need to search for alternative method of controlling antibiotic resistant pathogens. So we can see from here that by 2050, 1 person could die in every 3 seconds. This shows the danger of antibiotic resistance. Okay, fine. Now we come to metallic nanoparticles. Why we talk about metallic nanoparticles? Because they are well known as a promising antimicrobial agent. And we can see nowadays the metallic nanoparticles are used in large number of materials, including plastics, coating materials, foams, and also in fibers. And most importantly, the metallic nanoparticles have proven their effectiveness for treating infectious diseases, including antibiotic-resistant pathogen. So this is where the nanoparticles are important to be considered to treat the infections caused by antibiotic resistance pathogens. So among the metallic nanoparticles, zinc oxide, titanium dioxide, and silver nanoparticles are most commonly used in controlling the microbial growth. Remarkably, zinc oxide has greater advantage because of its highest photocatalytic efficiency and also the biocompatibility, means it's less toxic to human cells. And also, zinc oxide nanoparticles are efficient at multi-drug resistant bacteria treatment. Actually, you can see what's the mechanism, how the zinc oxide nanoparticles can kill the bacteria. Zinc oxide nanoparticles kill bacteria through the production of ROS, reactive oxygen species, that will cause the destruction of cellular components, including DNA, proteins, and also the cell membrane. Further, the contact of zinc oxide nanoparticles on the surface, on the cell wall of bacteria, can cause rupture of cell membrane, which in turn cause influx of more zinc ions and particles into the intracellular compartment, leading to oxidative stress. So basically, these are the three mechanisms responsible for killing the bacteria by zinc oxide nanoparticles. First one is by the production of reactive oxygen species. Second one, the release of zinc ions and entering into this is the same index which confirm the zinc oxide nanoparticles, the presence of zinc and oxygen. Now, we talk about Streptococcus pyogenes. We know Streptococcus pyogenes is gram-positive cocci arranged in chains. We can see from the picture. So it is well known for causing rheumatic heart disease. So and also it causes skin and wound infections. So we can see in our investigation, in our study, the number of colonies are reduced. We can see the comparison here. This is the 10 ppm nanoparticles, zinc oxide nanoparticle. Here is 50 and here 100 ppm of zinc oxide nanoparticle. So we can see here the number of colonies are reduced as the concentration of zinc oxide nanoparticles increased. So here we can see the negative control without any particle. We can see the number of colonies 181. For 10 it is 102, 50, 18. For 100 it is 4 colonies. And the positive control, no growth. So it shows the zinc oxide nanoparticles causes growth inhibition in a dose dependent manner. That means when we increase the concentration of nanoparticles and the growth inhibition also increased. Okay, so we can see the growth inhibition based on the colony count. So we reported a significant growth inhibition at 10, 50, and 100. So from 10 we can see there is a significant growth inhibition by zinc oxide nanoparticles. So in the turbidity method, we can see here the turbidity has decreased. There is a negative control. We can see as the concentration of zinc oxide nanoparticle increased, the turbidity also decreased. We can see as the particle concentration increases, the turbidity decreased. Again, the growth inhibition, we can see that the significant growth inhibition happened from 10 ppm of nanoparticles at 24 hours. Yes, so also you can see the growth inhibition, the turbidity method as well as by the colony count method. So why we want to do the both? Because the turbidity method will indicate the potential bacteriostatic effect of the nanoparticle nanoparticles, whereas the colony method indicates the bactericidal effect of the nanoparticles. So, to demonstrate the underlying mechanism of growth inhibition, we tested ROS, reactive oxygen species, produced by the treatment of zinc oxide nanoparticles. So, we reported significant increase in the production of ROS from 10 ppm of zinc oxide nanoparticles. So, further, ROS production eventually will lead to lipid peroxidation. This is the base for cell membrane rupture and killing of microorganisms. So we tested the lipid peroxidation also. You can see we reported a significant increase in lipid peroxidation from 10 ppm onwards, and also can damage the cell membrane. So that's why we used FTIR to identify the functional groups which are responsible for binding zinc oxide nanoparticles over the cell surface. So why we did all these things? So first thing, we tested the growth inhibition. Yeah, we reported significant growth inhibition even at the lowest concentrations of zinc oxide nanoparticles. To demonstrate the basic mechanism behind the growth inhibition, we reported ROS and also we reported lipid peroxidation. So, because of increase in reactive oxygen species and the subsequent lipid peroxidation, these are the basis for killing the bacteria. Okay. So now we tested the another bacteria, Proteus vulgaris. We know Proteus vulgaris are well known for urinary tract and wound infections. Basically, Proteus vulgaris is a gram-negative bacillus. So we can see here, similar with the structure of pyogens, we can see the number of colonies reduced as the concentration of zinc oxide nanoparticles increased. Then the similar to Streptococcus, we reported dose-dependent growth inhibition in Proteus vulgaris also, in both the methods, in turbidity as well as in colony count method. Yes, always also increased. But here we can see at 10 ppm we did not report significant ROS production. From 50 only we reported the ROS production. So similar to ROS lipid peroxidation also. So we can see here Streptococcus pyogen comparatively more sensitive than the Proteus vulgaris. So next, the bacteria we tested is Pseudomonas aeruginosa. We know Pseudomonas aeruginosa is well known for hospital acquired infections and for wound and soft tissue infections. So again, we can see the number of colonies are decreased as the concentration of nanoparticles increased. The similar report obtained in turbidity method also. Under the scanning electron microscopy, we can see the damage of bacterial cell surface. We can see like cell membrane rupture, cell distortion, or by the action of zinc oxide nanoparticles. On immunocompromised individuals. So here actually also same. We reported decrease in the bacterial, Saccharomyces cerevisiae turbidity as the concentration of zinc oxide nanoparticles increased. And the same thing we reported the significant growth inhibition of the Saccharomyces cerevisiae from 5 ppm of zinc oxide nanoparticles. So FTIR demonstrates the involvement of lipid, carbohydrate, proteins in the cellular binding of nanoparticles. So we can see here, typically we can see invagination on the surface of the yeast cell caused by the zinc oxide nanoparticles. And again, we can see the deformity and also the rupture, membrane rupture. Yes, you can see here the cell wrinkling, the rupture of cell membrane again. Yes, so actually, so we have so many organisms. I just showed you few bacteria and also one yeast cell. So apart from the bacterial yeast cell, actually we work on cyanobacteria, and we reported significant growth inhibition on cyanobacteria that we published in Q1 journal, US-based journal, and also we worked on Hematococcus pluvialis. That also we reported significant accumulation of nanoparticles and also significant growth inhibition by zinc oxide. And the projects that we are investigating the cytotoxic effects of metallic nanoparticles and the subsequent impact on nutritional values of nutrient care.
Evidence-based healthcare is nothing but an integration of best research practices as evidence for a drug candidate with clinical expertise and patient preference and patient values. So, what is the need for this evidence-based medicine in Ayush formulation? As we know, India has an ancient knowledge of traditional medicine. It contains a wealth of learning on information about today's well-being. It also plays an important role in meeting the global healthcare needs. So we are having a rich sources of medical knowledge in our country, but whether it is globally accepted or not. So that is why this evidence-based healthcare research is required in Ayush formulation. Even mainstreaming Indian system of medicine is one of the key vision of Government of India. So the lacunae are Ayush formulation are found to be highly effective, no doubt in that, and it is being used successfully for centuries, no doubt in that. But what is lacking? Meticulous experimentation in the drug standardization, clinical trials, and based on the efficacy, how many products has been developed with international standards. So these are the lacunae in the Ayush formulation. Hence, evidence-based scientific approach are the need of the hour to prove its efficacy for global acceptance. Okay, now I understood that Ayush formulation has to be standardized. Who are these stakeholders? The hero of this drug standardization are biomedical scientists. Along with biomedical scientists, clinicians has to join hand. Ultimately, each and every formulation has to be documented scientifically, and it has to be utilized for the common people. Here, the major role is played by the biomedical scientist along with the clinicians. So, any scientist, before starting this documentation process in the Ayush system of medicine, they have to follow certain golden rules. They have to choose a particular drug, a specific drug, and they have to measure the key parameters and what are the key attainable has to be documented. All those documentation has to be relevant to the particular type of disease. And one more thing it has to be followed is all this documentation has to be done in a time-bound manner. Hence, they can achieve their goal in a very easy way. So either they have to choose external drug or internal drug or documenting any clinical conditions based on the Ayush system or any diagnostic marker based on the Ayush system. They have to be more specific and also it has to be more relevant. So that is the first step for any scientist, those who are interested to work on the Ayush formulation. Because there are 32 types of internal medicine in Ayush system of medicine, starting from Surasam, Charu, Kudineer, which is nothing but a decoction, till Kuligai. But among these 32 types of internal medicine, the commonest things are either it is in a bioactive compounds. Okay, this is the second step. First, physical properties we are analyzing. Next, chemical properties. Next step is isolation of bioactive compounds to prove. So this is the compound which is play a role in a particular type of disease. However, if the drug is an oil, culture research facility before going for animal as well as human. Especially if a drug candidate is a cancer candidate, anti-cancer drug. The drug has to be tested initially in the cell line, in the cell culture research facility, and its efficacy has to be documented. It can be rat, mouse, or zebrafish, or even genetically engineered experimental animals. So after finding a good results, a strong positive results in the animal studies, it has to be tested for. It has to be tested with the human as a part of that's in the final stage of clinical trial. Okay. So in human studies, again, after giving the drug, its microscopical analysis, biochemical analysis, molecular biology, genetics, genomics, proteomics, metabolomics has to be that is the parameters related to the gene, parameters related to the genomics, biochemistry, all the sugar values from basic sugar values till the high-end values has to be documented. After documenting all these parameters, next we have to check for the pharmacokinetics of the drug candidate and the rate of absorption, how far the drug is absorbed, how far the drug is distributed, how far it is metabolized, what is its toxic effect. We have to check it. Then we have to document bioactivity and bioavailability. Finally, its therapeutic exploration has to be done. So from the physical property till the therapeutic exploration has to be done as a complete process for the global acceptance. So that is from the preclinical clinical research, final data analysis. After documenting all the analysis, these parameters has to be done. Next, so we have to check approve the particular drug kinetics of the drug candidate, regulate the rate of absorption, how far the drug is absorbed. I'm sure the drug is it will be accepted by the metabolized. What is its toxic effect? To check it. Then we have to document the drug is bioactivity and bioavailability accepted by the international community. Therapeutic exploration has to be done. Hybrid molecules with the from the physical joining of hybrid molecules or isolation of the herbs be done. We can create a synthetic drugs based on the herbs for the global, which is next step. So that is from the preclinical. So clinical research, final data from that known formulation after documenting all the efficacy, safety, route done. Next, from the known formulation, a synthetic product particular drug discovered, that is the ultimate of the candidate regulator rate of absorption. How far after drug discovery, when it is beneficial for the drug is it will be accepted by the metabolized. So from the what is its basic effect? New drug synthesis. We have a major role drug in bio-Ayush formulation research. Example, these are products finally by hence from the RNA for hybrid molecules with the from the physical joining of hybrid molecules or isolation of the herbs. So like this, we can create a synthetic drugs based on every drug for the present in the oil formulation has to come out as a step. So that is the product development candidate. So our candidate final data from the that is the analysis after vision and we have already as a scientific as well as the community have to check to work together upon this particular formulation as a product candidate regulator as well as we have to take after discovery to the international beneficial drug it will be accepted by this metabolized take home message. So from biomedical scientists and clinicians has to determine Ayush formulation what is its basic objective synthesis. There is a drug is Ayush formulation. It is research effective for clinical condition. Pharmaceutical companies are trends that call upon facing various challenges from the physical as a beneficial drug to humans. So like this, we can create a drug has to be documented scientifically developed as a commercial product. So that is pharmaceutical company for the product clinical candidate. So our candidates with this competing from the presentation that is the analysis. I thank Dr. and we have already opportunity to present this as a scientific as well as community. I want to work together upon the management of my institute, checking all the formulation as a product candidate as well as presentation. So have to take discovery all the international beneficial displayed only accepted by the so the take home message. So from once again, I thank you all. Thank you for listening. With this formulation is basic objective. The century has a drug is Ayush formulation. It is research. Effective for clinical condition. Pharmaceutical
Department of Biochemistry.
As a beneficial
Today I'm discussing about the effect of short chain fatty acids.
So like this,
so the cancer. Cancer is the proliferation of cells. So when we control the process, the spreading of cancer cells decreases. So there are a lot of dietary products we are taking to control all these. But among that, we must be very less know about the short chain fatty acids. It is actually the simplest process where the dietary fiber fragment and it's short chain fatty acids. But in our lifestyle, due to a lot of junk foods as well as non-vegetarian the vegetarian food items we take so that dietary fiber decreases. We are reducing the intake of short chain fatty acids. So that may be one of the reasons to proliferate the abnormal cells. So here, how do you get the short chain fatty? Very simple. These are the various dietary food when you take, which is mainly the undigested carbohydrates is the major source. So where we call cellulose, hemicellulose. Like the flora, especially anaerobic bacteria, and produce short chain fatty acids. So these short chain acids, Department of Biochemistry, they are acetic acid, propionic acid, and butyric acid. There is a 2 carbon, 3 carbon, and 4 carbon compounds. The 4 carbon compound is an important fatty acid. The food source which you can increase in our diet are legumes, effect of short chain fatty acids, barley, chia, broccoli, carrot, or sweet potato, very famous, onions, palms, bananas, apples, all these. So when we come to get the lot of short chain fatty acids, so the water deficiencies in our body, they can be oxidized to provide. There are a lot of and have also been shown to affect the immune system, cholesterol metabolism. You must be very less known about a direct link between short chain fatty acid, and simple inflammatory bowel cirrhosis, where the and controlled by intake of short chain and short chain that has a considerable in our lifestyle. A diet high in fat has been considered cancer. High fat means the lung while increased fiber and complex carbohydrates in the diet give the diet cancer. Butyric is believed to be mostly responsible for the tumor inhibitors, so that may be one of dietary fiber. Sodium butyrate is known to be an inhibitor of cell differentiation. It is believed that the protective effect of dietary fiber is associated with butyrate. So, how do you get the short chain the occurrence of these are the neoplasia in colonocysts. Various dietary example when you take associated with high butyrate protective against cancer. So this is the one of the mechanism how the short chain fatty acid control the short chain fatty acids intake increase, especially absorbed by simple diffusion, and it is associated with the ionic concentration of so when the diffuse short chain fatty acids stimulate the P21 gene, which is responsible for the cell cycle 4 carbon compounds. The 4 carbon suppresses and cell cycle get arrested and important food or in other way you can increase in our diet effect of short chain fatty acids get inhibited carrot cyclist or sweet potato sun and that will onions palms is controlled and inhibition bananas so when we come to get the lot of short chain the propionate or the water that binds into the plasma membrane, they can be OX3 signal protein coupled receptor. Provide and have domains. G protein domains. They can immune system immediate cell signaling event activation cholesterol metabolism and then cell cycle. That is direct short chain fatty acid accelerated. Cyclin D3 and simple is water bowls down regulation and so all these will control the proliferation of short chain fatty acids. Once the short chain fatty acid has been as cell cancer, high fat means the lung cancer, In the cancer cells, there
Which
is
is
no damage
the
under mitochondria. The vegetarian
so
to remove fiber, mitochondria, damaged mitochondria, mechanism by removing the glutamate is believed to be mostly responsible for the tumor inhibition.
Thank you for
That may be one of dietary mitochondria. Sodium citrate has been diffused into the cells to be an enzyme proliferator of the mitochondrial membrane potential and causes defect. The protective effect of dairy fiber is associated with production and control the occurrence of. So these are the aplasia, cytochrome C reductase and dietary factors. They will control the intake associated with high which will control apoptosis induction. And so this is the one of the mechanism how the method which control the abnormal fatty acid control the by this what we have understood is short chain fatty acids are the major byproducts of bacterial fermentation of undigested fibers in the colon, especially anaerobic circulation, and associated with the differentiation, growth arrest, and apoptosis in short chain So, can prevent the autophagy to retard mitochondrial defect or the in vitro studies demonstrate a role of short chain fatty acid in promoting autophagy and protecting the cells from apoptotic death. The relevance of this finding has yet to be suppresses. Moreover, and cell segment of colon cancer cells with short chain fatty acids and inhibitor of autophagy may be an effective therapy approach to be used. Other way, so overall, what I effect of short chain explain is we are less given importance to the short chain fatty acids and intestinal microbiota, which are very, very important to ferment sweet potato and produce short chain fatty acids. And that short chain fatty acids helps to protect various metabolic cell proliferation. So actually at present we take a lot of antibiotic for various different kinds of so the antibiotic will damage all the microbiota of the intestine where supposed to ferment the dietary fiber and gives you a lot of short chain fatty acids which is very good required for the health. We are stopping by the plasma membrane. That is why syndrome is very common nowadays because of the irradiation of the novel microbiome. By this, I request all of us to know about the importance of novel microbiota in the intestines, dietary fiber, and short chain fatty acid production. Cancer cell line growth as well as for the metabolic activation, cholesterol metabolism, cell cycle. That is which is short chain fatty acid, it is accelerated. Cyclin D3 and simple is water bowls down regulation and research so all these will control the proliferation and diet has a considerable a diet high in fat. Once the short chain fatty acid has as well as cell cancer. High fat means the lung cancer. In the cancer cells, there
Great
is no
use of
damage to the mitochondria. The vegetarian type fiber mitochondria, damaging mitochondria, by removing the
Once again,
glutamate is believed to be mostly responsible for the tumor in
thank you for
that may be volatile in the mitochondria. Sodium short-chain fatty acid diffuse into the cells. Lower the mitochondrial membrane and causes defect. That the protective effect of dietary fibers is associated with production and control the production of the occurrence of. So these are the cytochrome C reductase and dietary factors. They will control the which will control apoptosis induction. And so this is the one of the mechanism how the method which control the abnormal fatty acid control the by this what we have understood is short chain fatty acids are the major byproducts of bacterial fermentation of undigested fibers in the colon, especially particularly and associated with the differentiation, growth arrest and apoptosis in short cells. So when the autophagy to retard mitochondrial defect in vitro studies demonstrate a role of short-chain fatty acid in promoting autophagy cell cycle, protecting the cells from apoptotic death. The relevance of this finding has yet to be validated in one. Moreover, and cell segment of colon cancer cells with short-chain fatty acids and inhibitor of autophagy may be an effective therapy approach to be used. Other way, so overall, what I effect of short-chain explain is we are less given importance to the short-chain fatty acids and interstitial microbiota, which are very, very important to ferment sweet potato and produce short-chain fatty acids. And that short-chain fatty acids helps to protect various metabolic cell proliferation. So actually at present we take a lot of antibiotic for various different kinds of so the antibiotic will damage all the microbiota of the intestine where supposed to ferment the dietary fiber and gives you a lot of short-chain fatty acids, which is very good required for the health. We are stopping by the plasma membrane. That is why Down syndrome is very common nowadays because of the irradiation of the novel microbiome. By this, I request all of us to know about the importance of novel microbiota in the intestines and fiber and short-chain fatty acid production. Cancer cell line growth as well as for the metabolic activation, cholesterol metabolism, cell cycle. That is which is short-chain fatty acid, it is accelerated. Cyclin D3 simple is what about down regulation and cancer so all these will control the proliferation and short-chain fatty acids. One time, a diet high in fat. Once the short-chain fatty acid has as well as cell cancer. High fat means the lung cancer. In the cancer cells, there is no damage of mitochondria. So to remove fiber, mitochondria, damaged mitochondria, by removing the glutamate is believed to be mostly responsible for the tumor inhibition.
Thank you for
That may be one of the mitochondria. Sodium short-chain has been diffused into the cells. The mitochondrial membrane potential and causes defect. The protective effect of dietary fiber is associated with production and control the production of the occurrence of. So these are the cytochrome C reductase and dietary factors. They will control the which will control apoptosis induction. So this is the one of the mechanism how the method which control the short-chain fatty acid control the by this what we have understood is short-chain fatty acids are major byproducts of bacterial fermentation of undigested fibers in the colon, especially and associated with the differentiation, growth arrest and apoptosis in short-chain can prevent the autophagy to retard mitochondrial defect in vitro studies demonstrate a role of short-chain fatty acid in promoting autophagy and protecting the cells from apoptotic death. The relevance of this finding has yet to be suppresses. Moreover, and cell segment of colon cancer cells with short-chain fatty acids and inhibitor of autophagy may be an effective therapy approach to be used. Other way. So overall, what I effect of short-chain explain is we are less given importance to the short-chain fatty acids and intestinal microbiota, which are very, very important to ferment sweet potato and produce short-chain fatty acids. And that short-chain fatty acids helps to protect various metabolic cell proliferation. So actually at present we take a lot of antibiotic for various different kinds of so the antibiotic will damage all the microbiota of the intestine where supposed to ferment the dietary fiber and gives you a lot of short-chain fatty acids which is very good required for the health. We are stopping by the plasma membrane. That is why syndrome is very common nowadays because of the irradiation of the novel microbiome. By this, I request all of us to know about the importance of novel microbiota in the intestines, fiber, and short-chain fatty acid production. Cancer cell line growth as well as for the metabolic activation of cholesterol metabolism. Cell cycle that is which is short-chain fatty acid, it is accelerated. Cyclin D3 and simple is what about down regulation and process so all these will control the proliferation and that has a considerable a diet high in fat. Once the short-chain fatty acid has as well as cell cancer. High fat means the lung cancer. In the cancer cells, there
Features
is no
of
damage
the
mitochondria. The vegetarian
so
increased type fiber mitochondria, damaged mitochondria by removing the glutamate is believed to be mostly responsible for the tumor inhibition. That may be one in the mitochondria. Sodium bitarate has been diffused into the cells. Lower the mitochondrial membrane potential and causes defect. The protective effect of dietary fibers is associated with production and control the occurrence of. So these are the cytochrome C reductase and dietary factors. They will control the intake associated with high pituitary, which will control apoptosis induction. So this is the one of the mechanism how the method which control the abnormal fatty acid, control the by this what we have understood is short chain fatty acids are the byproducts of bacterial fermentation of undigested fibers in the colon, especially and associated with the differentiation, growth arrest and apoptosis in short chain when the autophagy to retard mitochondrial defect acetic acid. In vitro studies demonstrate a role of short chain fatty acid in promoting autophagy and protecting the cells from apoptotic death. The relevance of this finding has yet to be investigated in one. Moreover, and cell segment of colon cancer cells with short chain fatty acids and autophagy may be an effective therapy approach to be used. Other way, so overall, what I effect of short chain explain is we are less given importance to the short chain fatty acids and intestinal microbiota, which are very, very important to ferment sweet potatoes and produce short chain fatty acids. And that short chain fatty acids helps to protect various metabolic cell proliferation. So actually at present we take a lot of antibiotic for various different types of so the antibiotic will damage all the microbiota. So when where supposed to ferment the dietary fiber and gives you a lot of short chain fatty acids, which is very good required for the health. We are stopping by the plasma membrane. That is why Warm syndrome is very common nowadays because of the irradiation of the novel microbiome. By this, I request all of us to know about the performance of novel microbiota in the intestines, dietary fiber, and short chain fatty acid production. Cancer cell line growth as well as for the metabolic activation, cholesterol metabolism, cell cycle. That is which is short chain fatty acid, it is accelerated. Cyclin D3 is what about down regulation and so all these will control the proliferation and that has a considerable a diet high in fat. Once the short chain fatty acid has as well as cell cancer. High fat means cancer. In the cancer cells, there is no damage to the mitochondria. The vegetarian source type fiber mitochondria, damaged mitochondria is balanced by removing the glutamate is believed to be mostly responsible for the inversion. That may be volatile in the mitochondria. The short-chain fatty acid diffuse into the cells. Lower the mitochondrial membrane potential and causes defect. The protective effect of dietary fibers is associated with production and control the production of the occurrence of. So these are the cytochrome C reductase and dietary factors. They will control the which will control apoptosis induction. And so this is the one of the mechanisms how the method which control the abnormal fatty acid control the by this what we have understood is short chain fatty acids are the major byproducts of bacterial fermentation of undigested fibers in the colon, especially and associated with the differentiation, growth arrest, and apoptosis in short cells. When the autophagy to retard mitochondrial defect in vitro studies demonstrate a role of short-chain fatty acid in promoting autophagy and protecting the cells from apoptotic death. The relevance of this finding has yet to be studied in one. Moreover, cell segment of colon cancer cells with short-chain fatty acids and inhibitor of autophagy may be an effective therapy approach to be used. So overall, what I effect of short-chain explain is we are less given importance to the short-chain fatty acids and intestinal microbiota, which are very, very important to ferment and produce short-chain fatty acids. And that short-chain fatty acids helps to protect various metabolic cell proliferation. So actually at present we take a lot of antibiotic for various different types of so the antibiotic will damage all the microbiota of the intestine where supposed to compress the dietary fiber and gives you a lot of short-chain fatty acids, which is very good required for the health. We are stopping by the plasma membrane. That is why Down syndrome is very common nowadays because of the irradiation of the novel microbiome. By this, I request all of us to know about the importance of novel microbiota in the intestines and fiber and short-chain fatty acid production. Cancer cell line growth as well as for the metabolic activation, cholesterol metabolism, cell cycle. That is which is short-chain fatty acid, it is accelerated. Cyclin D3 down regulation and so all these will control the proliferation and that has a a diet high in fat. Once the short-chain fatty acid has been cell cancer, high fat means the lung cancer. In the cancer cells, there is no damage to the mitochondria. So to remove fiber, mitochondria, damaged mitochondria, by removing the glutamate is believed to be mostly responsible for the tumor inhibition.
Thank you for
That may be one of the mitochondria. Sodium short-chain has been diffused into the cells. The mitochondrial membrane potential and causes defect. The protective effect of dietary fiber is associated with production and control the production of the occurrence of. So these are the cytochrome C reductase and dietary factors. They will control the intake which will control apoptosis induction. So this is the one of the mechanism how the method which control the short-chain fatty acid control the by this what we have understood is short-chain fatty acids are major byproducts of bacterial fermentation of undigested fibers in the colon, especially and associated with the differentiation, growth arrest and apoptosis in short-chain So can prevent the autophagy to retard mitochondrial defect The in vitro studies demonstrate a role of short-chain fatty acid in promoting autophagy and protecting the cells from apoptotic death. The relevance of this finding has yet to be suppresses. Moreover, and cell segment of colon cancer cells with short-chain fatty acids and inhibitor of autophagy may be an effective therapy approach to be used. Other way. So overall, what I effect of short-chain explain is we are less given importance to the short-chain fatty acids and intestinal microbiota, which are very, very important to ferment sweet potato and produce short-chain fatty acids. And that short-chain fatty acids helps to protect various metabolic cell proliferation. So actually at present we take a lot of antibiotic for various different so the antibiotic will damage all the microbiota of the intestine where supposed to ferment the dietary fiber and gives you a lot of short-chain fatty acids which is very good required for the health. We are stopping by the plasma membrane. That is why syndrome is very common nowadays because of the irradiation of the novel microbiome. By this, I request all of us to know about the importance of novel microbiota in the intestines, fiber, and short-chain fatty acid production. Cancer cell line growth as well as for the metabolic activation of cholesterol metabolism. Cell cycle. That is which is short-chain fatty acid, it is accelerated. Cyclin D3 and simple is what about down regulation and process so all these will control the proliferation and that has a considerable the short-chain fatty acid has been as cell cancer. High fat means the lung cancer. In the cancer cells, there
Nature
is no
has
damage
damage
to the mitochondria. The vegetarian
So
includes type fiber, mitochondria, damage in mitochondria, mechanisms by removing the glutamate is believed to be mostly responsible for the tumor inhibition. That may be one in the mitochondria. Sodium butyrate has been diffused into the cells. Lower the mitochondrial membrane potential and causes defect. That the protective effect of dietary fibers is associated with production and control the occurrence of. So these are the cytochrome C reductase and dietary fatty filters. They will control the intake associated with high pituitary, which will control apoptosis induction. And so this is the one of the mechanism how the method which control the abnormal fatty acid, control the by this what we have understood is short chain fatty acids are the major byproducts of bacterial fermentation of undigested fibers in the colon, especially anaerobic particulate and associated with the differentiation, growth arrest and apoptosis in short chain when the autophagy to retard mitochondrial defect acetic acid, the in vitro studies demonstrate a role of short chain fatty acid in promoting autophagy and protecting the cells from apoptotic death. The relevance of this finding has yet to be validated in one. Moreover, and cell segment of colon cancer cells with short chain fatty acids and inhibitor of autophagy may be an effective therapy approach to be used. Other way, so overall, what I effect of short chain explain is we are less given importance to the short chain fatty acids and intestinal microbiota, which are very, very important to ferment sweet potatoes and produce short chain fatty acids. And that short chain fatty acids helps to protect various metabolic less cell proliferation. So actually at present we take a lot of antibiotic for various different types of so the antibiotic will damage all the microbiota. So when where supposed to ferment the dietary fiber and gives you a lot of short chain fatty acids, which is very good required for the health. We are stopping by the plasma membrane. That is why irradiation syndrome is very common nowadays because of the irradiation of the novel microbiome. By this, I request all of us to know about the performance of novel microbiota in the intestines and dietary fiber and short chain fatty acid production. There is the cancer cell line growth as well as for the metabolic activation, cholesterol metabolism. Cell cycle. That is which is short chain fatty acid, it is accelerated. Cyclin D3 down regulation and research so all these will control the proliferation and short chain fatty acids. A diet high in fat. Once the short chain fatty acid has as well as cell cancer. High fat means cancer. In the cancer cells, there is no damage to the mitochondria. The vegetarian source type fiber mitochondria, damaged mitochondria is by the mechanism by removing the glutamate is believed to be mostly responsible for the tumor that may be volatile in the mitochondria. Sodium citrate has been diffused into the cells. Lower the mitochondrial membrane potential and causes defect. The protective effect of dietary fiber is associated with production and control the production of the occurrence of. So these are the cytochrome C reductase and dietary factors. They will control the which will control apoptosis induction. And so this is the one of the mechanisms how the method which control the abnormal fatty acid control the by this what we have understood is short chain fatty acids are the major byproducts of bacterial fermentation of undigested fibers in the colon, especially and associated with the differentiation, growth arrest and apoptosis in short cells when the autophagy to retard mitochondrial defect in vitro studies demonstrate a role of short-chain fatty acid in promoting autophagy and protecting the cells from apoptotic death. The relevance of this finding has yet to be studied in one. Moreover, cell segment of colon cancer cells with short-chain fatty acids and inhibitor of autophagy may be an effective therapy approach to be used. So overall, what I effect of short-chain explain is we are less given importance to the short-chain fatty acids and intestinal microbiota, which are very, very important to ferment and produce short-chain fatty acids. And that short-chain fatty acids helps to protect various metabolic cell proliferation. So actually at present we take a lot of antibiotic for various different types of so the antibiotic will damage all the microbiota of the intestine where supposed to compress the dietary fiber and gives you a lot of short-chain fatty acids, which is very good required for the health. We are stopping by the plasma membrane. That is why Down syndrome is very common nowadays because of the irradiation of the novel microbiome. By this, I request all of us to know about the performance of novel microbiota in the intestines and fiber and short-chain fatty acid production. There is cancer cell line growth as well as for the metabolic activation, cholesterol metabolism, cell cycle. That is which is short-chain fatty acid, it is accelerated. Cyclin D3 down regulation and process so all these will control the proliferation and that has a a diet high in fat. Once the short-chain fatty acid has as well as cell cancer. High fat means the lung In the cancer cells, there is no damage to the mitochondria. So to remove fiber, mitochondria, damaged mitochondria, by removing the glutamate is believed to be mostly responsible for the tumor inhibition. That may be one of in the mitochondria. Sodium short-chain fatty acid diffuse into the cells. The mitochondrial membrane potential and causes defect. The protective effect of dietary fiber is associated with production and control the production of the occurrence of. So these are the cytochrome C reductase and dietary factors. They will control the which will control apoptosis induction. So this is the one of the mechanism how the method which control the short-chain fatty acid, control the by this what we have understood is short-chain fatty acids are the major byproducts of bacterial fermentation of undigested fibers in the colon, especially anaerobic fermentation, and associated with the differentiation, growth arrest, and apoptosis in short-chain cells of the future. So, can prevent the autophagy to retard mitochondrial defect or in vitro studies demonstrate a role of short-chain fatty acid in promoting autophagy and protecting the cells from apoptotic death. The relevance of this finding has yet to be suppresses. Moreover, and cell segment of colon cancer cells with short-chain fatty acids and inhibitor of autophagy may be an effective therapy approach to be used. Other way. So overall, what I effect of short-chain explain is we are less given importance to the short-chain fatty acids and intestinal microbiota, which are very, very important to ferment sweet potato and produce short-chain fatty acids. And that short-chain fatty acids helps to protect various metabolic cell proliferation. So actually at present we take a lot of antibiotic for various different kinds of so the antibiotic will damage all the microbiota of the intestine where supposed to ferment the dietary fiber and gives you a lot of short-chain fatty acids, which is very good required for the health. We are stopping by the plasma membrane. That is why Stevens-Jones syndrome is very common nowadays because of the irradiation of the novel microbiome. By this, I request all of us to know about the importance of novel microbiota in the intestines, fiber, and short-chain fatty acid production. Cancer cell line growth as well as for the metabolic activation of cholesterol metabolism. Cell cycle. That is short-chain fatty acid was accelerated. Cyclin D3 and simple is water bowls down regulation and process so all these will control the proliferation and that has a considerable the short-chain fatty acid has been as cell cancer. High fat means the lung cancer. In the cancer cells, there
There
is no
is
damage
no
to the
mitochondria.
mitochondria. The vegetarian
So
includes type fiber, mitochondria, damaged mitochondria, mechanism by removing the glutamate is believed to be mostly responsible for the tumor that may be volatile in the mitochondria. Sodium butyrate has been diffused into the cells to be an proliferation of the mitochondrial membrane potential and causes defect. That the protective effect of dietary fibers is associated with production and control the occurrence of. So these are the cytochrome C reductase and dietary factors. They will control the intake associated with high pituitary, which will control apoptosis induction. And so this is the one of the mechanism how the method which control the abnormal fatty acid, control the by this what we have understood is short chain fatty acids are major byproducts of bacterial fermentation of undigested fibers in the colon, especially anaerobic particulate and associated with the iron differentiation, growth arrest and apoptosis in short cells. So these short chain can prevent the autophagy to retard mitochondrial defect and in vitro studies demonstrate a role of short chain fatty acid in promoting autophagy and protecting the cells from apoptotic death. The relevance of this finding has yet to be validated in one. Moreover, and cell segment of colon cancer cells with short chain fatty acids and inhibitor of autophagy may be an effective therapy approach to be used. Other way, so overall, what I effect of short chain explain is we are less given importance to the short chain fatty acids and intestinal microbiota, which are very, very important to ferment sweet potatoes and produce short chain fatty acids. And that short chain fatty acids helps to protect various metabolic cell proliferation. So actually at present we take a lot of antibiotic for various different kinds of so the antibiotic will damage all the microbiota. So when we're supposed to ferment the dietary fiber and gives you a lot of short chain fatty acids, which is very good required for the health. We are stopping by the plasma membrane. That is why Down syndrome is very common nowadays because of the irradiation of the novel microbiome. By this, I request all of us to know about the performance of novel microbiota in the intestines, and fiber, and short chain fatty acid production. Cancer cell line growth as well as for the metabolic activation, cholesterol metabolism. Cell cycle. That is which is short chain fatty acid, it is accelerated. Cyclin D3 down regulation and research so all these will control the proliferation and that has a a diet high in fat. Once the short chain fatty acid has as well as cell cancer. High fat means the lung cancer. In the cancer cells, there is no damage to the mitochondria. The vegetarian foods type fiber mitochondria, damaging mitochondria is balanced by removing the glutamate is believed to be mostly responsible for the tumor inhibition. That may be one in the mitochondria. Sodium citrate has been diffused into the cells. The mitochondrial membrane potential and causes defect. The protective effect of dietary fiber is associated with production and control the production of the occurrence of. So these are the cytochrome C reductase and dietary factors. They will control the which will control apoptosis induction. And so this is the one of the mechanisms how the method which control the absorption fatty acid, control the by this what we have understood is short chain fatty acids are the major byproducts of bacterial fermentation of undigested fibers in the colon, especially anaerobic fermentation, and associated with the differentiation, growth arrest, and apoptosis in short cells. When the autophagy to retard mitochondrial defect
So ultimately, the marine algae are considered to be panacea for
the relevance of this
for driving cosmeceuticals as well as nutraceuticals, which can be renewable roses, new source of natural products which can be derived from the medicine and medicine by products. So let's consider the nutraceutical as well as cosmeceuticals as food as well as medicine. Thank you.
Supposed to the fiber and gives you a lot of short chain fatty acids. Is very good required for the health. We are stopping by the plasma membrane. That is why syndrome is very common nowadays because of the irradiation of the novel microbiome. By this, I request all of us to know about the causes of novel microbiota in the intestines, dietary fiber, and short chain fatty acid production. Cancer cell line growth as well as for the metabolic activation, cholesterol metabolism. Cell cycle. That is which is short chain fatty acid, it is accelerated. Cyclin D3 and simple is what about down regulation and process so all these will control the proliferation and that has a considerable a diet high in fat. Once the short chain fatty acid has as well as cell cancer. High fat means the lung cancer. In the cancer cells, there is no damage to the mitochondria. The vegetarian so to remove fiber, mitochondria, damaging mitochondria is by the mechanism by removing the nutrient is believed to be mostly responsible for the tumor that may be volatile in the mitochondria. Sodium short-chain fatty acid diffuse into the cells. To be an inhibit proliferation of the mitochondrial membrane potential and causes defect. That the protective effect of dietary fibers is associated with production and control the production of the occurrence of. So these are the cytochrome C reductase and dietary factors. They will control the which will control apoptosis induction. And so this is the one of the mechanisms how the said which control the abnormal fatty acid, control the by this what we have understood is short chain fatty acids are the major byproducts of bacterial fermentation of undigested fibers in the colon, especially anaerobic particulation, and associated with the differentiation, growth arrest, and apoptosis in short cells. So when the autophagy to retard mitochondrial defect
so ultimately, the marine algae are considered to be panacea for
the relevance of this
for driving cosmeceuticals as well as nutraceuticals, which can be renewable roses, natural products which can be derived from the medicine and medicine diet by Hippocrates. So consider nutraceutical as well as cosmeceuticals as food as well as medicine. Thank you.
Inhibition antibiotics will damage all the mitochondria. Supposed to ferment the dietary fiber and gives you a lot of short chain fatty acids. This is very good required for the health. We are stopping by the plasma membrane. That is why Down syndrome is very common nowadays because of the irradiation of the novel microbiome. By this, I request all of us to know about the importance of novel microbiome and the intestines and fiber and short chain fatty acid production. Cancer cell line growth as well as for the metabolic activation, cholesterol metabolism, cell cycle. That is which is short chain fatty acid was accelerated. Cyclin D3 simple is what about down regulation and process so all these will control the proliferation and that has a the short chain fatty acid has as well as cell cancer. High fat means the lung cancer. In the cancer cells, there
Creatures
is no
are
damage
damaged
to the mitochondria. The vegetarian
So
to remove fiber, mitochondria, damaged mitochondria, mechanisms by removing the
once again,
it is believed to be mostly responsible for the tumor in
thank you for
that may be
determining
volatile in the mitochondria. Sodium bitarate has been diffused into the cells. To be an inhibitor of the mitochondrial membrane potential and causes defect. That the protective effect of dietary fibers is associated with production and control the occurrence of. So these are the cytochrome C reductase and dietary factors. They will control the intake associated with high which will control apoptosis induction. And so this is the one of the mechanism how the method which control the abnormal fatty acid, control the by this what we have understood is short chain fatty acids are major byproducts of bacterial fermentation of undigested fibers in the colon, especially anaerobic particulate and associated with the iron differentiation, growth arrest and apoptosis in short cells. So can prevent the autophagy to retard mitochondrial defect in vitro studies demonstrated.
So ultimately, the marine algae are considered to be panacea for
the relevance of this
for driving cosmeceuticals as well as nutraceuticals, which can be renewable roses, natural products which can be derived from the medicine and medicine the digestion by the products. So let's consider derived nutraceutical as well as cosmeceuticals as food as well as medicine. Thank you.
Supposed to convert the dietary fiber and gives you a lot of short chain fatty acids, which is very good required for the health. We are stopping by the plasma membrane. That is why Down syndrome is very common nowadays because of the irradiation of the novel microbiome. By this, I request all of us to know about the importance of novel microbiome and the intestines, dietary fiber, and short chain fatty acid production. There is the cancer cell line growth as well as for the metabolic activation, cholesterol metabolism. Cell cycle. That is the which is short chain fatty acid accelerated. Cyclin D3 down regulation and process so all these will control the proliferation and that has a one time a diet high in fat. Once the short chain fatty acid has been cell cancer, high fat means the lung cancer. In the cancer cells, there is no damage of the mitochondria. So to remove fiber, mitochondria, damaged mitochondria, by removing the glutamate is believed to be mostly responsible for the tumor inhibition. That may be one of the mitochondria. Sodium citrate has been diffused into the cells. The mitochondrial membrane potential and causes defect. The protective effect of carrier fiber is associated with protection and control the occurrence of. So the
my name is Sri
these are the cytochrome C reductase is identified examples. They will control the intake associated with high which will control apoptosis induction. And so this is the one of the mechanism how the method which control the absorption fatty acid, control the by this what we have understood is short chain fatty acids are the major byproducts of fermentation of undigested fibers in the colon, especially and associated with the high differentiation, growth arrest and apoptosis in short chain when the autophagy to retard mitochondrial defect
So ultimately, the marine algae are considered to be panacea for
the relevance of this
cosmeceuticals as well as nutraceuticals, which can be renewable process,
members of IABM's family,
natural products which can be derived from
having International Virtual Conference of IABM, 41st Annual Conference of Biomedical Sciences, recent trends in biomedical research. The very country generosity of IABM and organized the 41st IABM Virtual Conference has been virtual and the edition in the last 4 to 6 weeks. The moral support of the President, Dr. D.K. Paul, and all the speakers, EC members, advisors, Dr. Venkatraman, Dr. Rajgopal, Dr. Gurunala Sundari, and all other officers, senior members, advisors, has wanted the Padma Madam's
that is why
well, during the last 3 days, we have had 12 international speakers, 20 national speakers, faculty, research scholars, and researchers by 15 scholars, postgraduates, and graduate students. Totally 185 deliberations. We had waived registration fees for research scholars and postgraduate and graduate students to encourage them to participate in the conference. We had a very good response because younger generation is more tech savvy when compared to me, older generation. For faculty also we had a nominal registration fee. I'm sure all of us had a great time interacting with the scientists from all over the world. Starting from USA, then Saudi Arabia, Pakistan, Sri Lanka, Malaysia, and all parts of the world. It was indeed a unique experience. Moreover, this COVID-19 has taught us how to communicate rather than commute to attend these conferences. With these few words, I heartily welcome one of the valedictorian. I welcome Dr. Sri Kumar Chakravarti, Director, Research and Innovation, to preside over this function.
I welcome Dr. E. Patel, General
In the
Secretary,
cancer cells,
IAPMS,
there
and
is a
Coordinating
mitochondrial
Secretary
conference
to announce the prizes to the winners of oral
mitochondrial
complex
mitochondria
nutrition Biomedical, one of the prize sponsors.
removing the
I also welcome Dr. Betty Selvan
mostly
Subramanian
responsible for the tumor immunity.
Thank you. Jai Hind.
Thank you, Dr.
That may be
Subramanian.
volatile in the mitochondria. Sodium citrate has been diffused into the cells. And causes defect. The protective effect of dairy fiber is associated with production and control the
And the role of genes in cancer, Mane. This is not only a revisiting, and I'm here to express a few, but also a very important how various types of genes can affect the possible outcome of the cancer in any way.
So this is the one of the mechanism how
Now I'm going
the
to start with a small quote and a poster from a movie that came about 10 years ago.
By this, what we have understood
And I'm sure you remember
short
this
chain
movie.
fatty acids
The
are
movie is
short
along
chain
with
fatty acids of
Now, as much as this movie is all
of
about external
undigested
mutants and
fibers
mutations
in
in
the
people,
colic increase,
what I'm going to talk today is a little bit about mutations
and
within our
associated with the
So I'm going to relate a lot of what I'm going to talk today to this
in
movie.
short chain
This movie is quite a fun fiction movie, but it's quite enjoyable. I'm not encouraging you to see this movie, but probably with a lot of genes with this
So ultimately, the marine
let's
organs
move on.
are
Now,
considered
many
to be the
slides
issue
say
for
proteins are survival. So that is
the
how it happens in the human body as well. As we keep growing,
cosmeceuticals as well as nutraceuticals, which can be
Good evening, respected international speakers, personal speakers,
renewed
chairpersons,
those
delegates, members of IAPMS family,
natural products which can be derived from
after having successfully completed the international virtual conference of IAPMS, the 41st annual conference of Future Association of Biomedical Scientists,
become expert
recent trends in biomedical research. Dr. E. Patil, General Secretary of IAPMS, has outlined several of the 41st IAPMS virtual conference has been working for its efficient delivery in the last 4 to 6 weeks. The moral support of the President, Dr. G.K. Paul, and all the senior EC members, advisors, Dr. Venkatraman, Dr. Rajagopal, Dr. Gurunala Sundari, and all other officers, senior members, advisors, has pointed for the Berlin Management Forum. Well, during the last 3 days, we have had 12 international speakers, 20 national speakers,
Now, before that, let me
faculty,
briefly
research
take you through
scholars,
how
and e-posters by
protein sits in our body. It is actually
15
nestled very perfectly
scholars,
in some
postgraduates, and graduate students.
When you zoom into the nucleus, you see the chromosomes.
Totally 125 deliberations
Which form an important constituent in the nucleus. Now let us slice the chromosome.
We had waived the registration fees for research scholars and
DNAs. Now DNA is the main backbone of what we are.
graduate
And you pick up the
students to encourage them to participate in the
and
conference.
genes are just very tiny components in the DNA. And
we had a very good response
response. Now, I've broken down
because
the
younger generation is more
so you
tech
can
savvy
imagine
than
how
compared to
small
me,
the
older generation.
nucleotide base pair is. It is even tinier than penicillin.
Cyclin
For
B3
faculty also, we had a nominal
Since it is so tiny, we need to realize that the receptor is identical in most cases. Now, for all of you who are listening to this talk of mine, I can guarantee that your DNA is identical, as is mine, but all differences are less than 0.1%.
And
So,
Saudi
this is
Arabia,
an example of
Sri
Iron
Lanka,
Man, Robert Downey
Malaysia.
Jr. and Sylvester Stallone. Both are totally different superheroes, actors in Hollywood, but almost all the DNA is the same.
And all
There is
parts
just
of
one
India.
small variation between
The unique
C and G or A and T, which is the difference between them, which makes them different people. And that is the difference between all the people among the 6 billion people in our planet. I know it's hard to believe sometimes, but that is the truth. It means all of us are almost the same. But the reason we are different is because of a very, very, very tiny break in the DNA.
With these few words,
So what are we looking
for?
Now, I'm sure all of you have smartphones and all of you have Google Maps.
I welcome
You would
Dr.
actually type the place
Shashri.
first, and then the Google Maps would first show you the whole country or the world at a distance. When you zoom out, and gradually as you zoom in, you can see the country, the address, and so on. The same thing is what we do in the human body. We are looking for a human genome. As you know, the human genome is big. We need to search for the chromosome, and then the band in the chromosome, and then the gene.
In the cancer cells, there is
a needle in a haystack. You know, a big haystack in farms. About 700, 800 cubic yards when you clump them together. And you're looking for one needle. One needle is about so that is what we are doing when we do a search for a DNA. It is so easy, but we have modern innovative methods to do a genomic.
In the mitochondria, sodium citrate has easily diffused into the cells.
Now, in the 4th edition of Molecular Biology of the Cell, DNA repair mechanisms. If humans lived long enough, they would all eventually develop. Now, what does this put? It means that if a person dies due to any reason other than a cancer, it is because the other disease overtaken otherwise, is meant to die from a cancer. Because of the rapid mutations that we keep getting in life, all of us and the role of genes, assuming that we did not have a disease due to lifestyle changes, a heart attack or an accident or diabetes or anything, we would all eventually die. Not only again, you go to the Bible. The Bible says, "Seek and you shall find." That's what and that's what many of the Italian Renaissance artists have said and shown in the pictures. Seek and you shall find. Which means that cancer find something, you have to search for it. The same thing goes in life as well. And the same thing goes for the DNA sequence as well. If you want to find it, that is what we aim to seek in this session. With a small code, but the genome sequence is one that came about 10 years ago. If I'm rich enough to do a genomic sequence, now I understand that we can do a genomic sequence for $200 US. And I'm sure you remember. I can prevent myself from these diseases, but no, that is not the answer. As much as this movie can cause a cancer. Is all about X-Men and mutants and mutations. There are different that can be altered to cause a cancer. Now I'm going to briefly discuss some basic fundamental concepts on so I'm going to relate a lot of what repair genes, anti-apoptotic genes, and methylation of genes. While methylation of genes is not actually a way to this a brake gets switched on or stopped, or a lock gets switched on or stopped. But it's quite enjoyable.
all have proto-oncogenes. These are normal genes that produce proteins and regulate normal cell proliferation, which means proto-oncogenes help
So
the body
ultimately, the biomarkers are considered to be studied
stimulate the normal growth. But at the same time,
or
they also
the
ensure that the growth is. That is how it happens in the human body as well. Now, let me give you the example of N. Sabanur, who was a normal Egyptian bad, and he turned into what we see as the apocalypse in the same movie that I told you in the beginning, X-Men Apocalypse. Oncogene. And the best example of a proto-oncogene is this man N. Sabanur. The role of a proto-oncogene is to stimulate the process whereby the growth factor meets with the growth factor receptor. The signal in the and the cells to proliferate. Now, there are numerous proto-oncogenes and genes, as I say. So, there's a different proto-oncogene that helps. It could be growth factor receptors, proto-oncogenes such as C6, CFB2, CRAS, CMYC, BCL2, and each of these has a different these are just some examples of proto-oncogenes that I'm telling you. There are numerous proto-oncogenes which are recorded up to date. If they are altered, it can increase the cell proliferation, or the cell proliferation can be made abnormal. So, cell proliferation anymore, the cell proliferation becomes abnormal. It can lead to a neoplastic proliferation or a cancerous proliferation. We need to know that the genes in our body. So, once a proto-oncogene is activated, it becomes a proto-oncogene, and an oncogene is a gene that is an active man becomes an abnormal mutant. So inside our body, the normal